I will now introduce our moderators for today. We have Dana from NOAA, as well as Caroline from Florida Sea Grant. So over to you to continue with your introductions.
Thank you, Fidela. Good morning, everyone. Thanks for joining us today for this special session on stony coral tissue loss disease. I'm Dana Mendez, and I'm with the U.S. National Oceanic and Atmospheric Administration, also known as NOAA, and I serve as the Atlantic Caribbean team lead for our Coral Reef Conservation Program. And I'm joined today by my colleagues Emma Doyle with GCFI and MPA Connect, as well as Caroline McLaughlin, who is the U.S. National Coral Disease Coordinator with Florida Sea Grant. Thanks to our partner and host, GCFI, for generously allocating this time at this year's conference to share recent developments with the region-wide outbreak of stony coral tissue loss disease, a disease event that is changing the face of Caribbean coral reef ecosystems. As you can see on the map here, we're showing all of the places where stony coral tissue loss disease as of today has now been confirmed. And along the left-hand side, you can see those places. I'd also like to thank our partners, Dr. Judy Lang and Dr. Patricia Kramer of the Atlantic and Gulf Rapid Reef Assessment, AGRA, both for producing this map, as well as their leadership in providing expertise and assistance across the region in tracking the spread of SCTLD and maintaining both a website and data dashboard that have served as a critical hub for reporting and information sharing on the disease event. So stony coral tissue loss disease is now being referenced as the most lethal coral disease ever recorded. It is impacting 27 stony coral species, including our major reef builders like the star and brain corals, as well as species that were already threatened with extinction before this new menace appeared, like the pillar coral. It was first observed in Miami in late 2014. And SCTLD has now been confirmed in the 20 different Caribbean countries and territories you see on the left-hand side of your screen. It moves rapidly across large reef areas, infecting susceptible species and killing entire colonies in a matter of weeks, leaving behind bare skeletons soon to be repopulated with algae. There's been a groundswell of effort over the past few years by scientists, resource managers, field practitioners, stakeholders, and conservationists in Florida and across the Caribbean to investigate this outbreak and to take action in an effort to minimize its impacts. 19 Caribbean countries and territories are actively monitoring the spread of SCTLD. 20 are conducting SCTLD outreach and education and engaging with the public and key stakeholders. And 11 countries and territories are intervening with treatments to prevent disease spread. We are here today to share some of the most recent science on SCTLD with you and to highlight a few of the many efforts to respond to this disease across the region. I'd now like to introduce Emma Doyle, GCFI board member and MPA Connect coordinator, who's briefly going to highlight some of the efforts we've been leading in the region to build capacity to respond to the threat of stony coral tissue loss disease. Emma?
Okay, thank you very much, Dana. Well, good morning, everyone. It's great to be here with you all. MPA Connect has actually worked since 2018 to build management capacity in relation to stony coral tissue loss disease. Our most recent efforts are actually really nicely summed up in this fabulous GCFI74 poster, which you can find online on the conference app. It's full of hyperlinks that take you directly to the things that I'm going to tell you about now. Now, there are four main areas of stony coral tissue loss disease capacity building efforts that we've worked on. There's been training, networking, we've created materials and resources, and we've provided some small grant support. Now, firstly, we've run several trainings about the disease. They've been both face-to-face and online, and there are great recordings available about disease ID, monitoring, and about response planning and intervention. So if you please go to the poster, you can follow the hyperlinks on it, or you can find those resources at the GCFI and at the AGRA website. Secondly, networking. Did you know that advice and expertise on stony coral tissue loss disease is no more than one step away from you? Please be aware that there's a Caribbean cooperation team about stony coral tissue loss disease that's jointly hosted by NOAA and AGRA. It meets monthly with regional organisations and every second month with a large group of managers from about 30 countries. So please let us know if you'd ever like to join that. MPA Connect is also helping managers to network and liaise with potential partners, especially in relation to treatment and in the newer area of coral rescue. We also coordinate a WhatsApp group about action on stony coral tissue loss disease. Now, thirdly, there are lots of materials and resources available online that you can use and that you can use to train others. Many of these are available in English, in Spanish, and in French. Now, there's an online dashboard and mapping tool, as Dana just showed. There are posters on disease ID, prevention and reporting. There's roving diver survey sheets online. There's a series of fact sheets about many aspects of stony coral tissue loss disease, including things like the use of antibiotics in treatment. And there's practical videos with tips for fishers, divers, information on monitoring protocols, a video guide on data analysis, and practical tips on disease treatment. Now, lastly, I can't mention resources without referring to much needed funding. MPA Connect is helping Caribbean managers with small grants for local stony coral tissue loss disease outreach and monitoring, and for exchange visits. So please stay tuned for further small grant opportunities, and please give MPA Connect a follow on your favourite social media. Now, these capacity building efforts are the result of many minds coming together and sharing about what's needed and about what's been learned. So I'd like to give a big thank you to everyone who so willingly and generously shares with our network. So I'll now hand over to Carolyn McLaughlin to give you an overview of this morning's session.
Hello, good morning, everybody. My name is Carolyn McLaughlin, and I'm the National Coral Disease Coordinator working with the University of Florida, Florida Sea Grant. So today our session is going to be broken into two parts. The first session will focus in on research and innovations, and then the second series of presentations will focus on response and intervention. So at the conclusion of each separate session, we will have a question and answer session of about 20 or 25 minutes. And so as you're listening to presentations, please go ahead and enter any questions you may have into the chat. And then Emma will share your questions with presenters during the live Q&A session after the presentation. Speakers, a reminder that you do have 8 minutes each for your presentations. Dana will be giving you a 2-minute warning near the end of your presentation. So on that note, I think we are good to go. We are going to start in with a research session, and our first presentation this morning is by Aldo Croquer with the Nature Conservancy, focused on the status of stony coral tissue loss disease across the northern coast of the Dominican Republic. Okay, so we should be good to go, Aldo.
Thank you so much. Hello, everyone, and thank you for joining us today. So today I'm going to give you a summary of the status of this disease in the northern coast of the Dominican Republic. Next one, please. So as for specific objectives and background, I want to tell you that stony coral tissue loss disease was first reported in 2019 in Cayo Arenas, Punta Cana, northwest of the Dominican Republic. In 2020, Red Arrecifal Dominicana and TNC designed and published a national action plan to tackle down the problem. In 2021, we started the monitoring program, and is currently being implemented in 3 regions, 3 localities per region, and at least 3 sites per location. In total, we have more than 27 sites that have been monitoring since April and May, August, and July of this year. Next one, please. So the objective of my talk is to present what we really know about stony coral tissue loss disease in the Dominican Republic, to present spatial patterns observed across the northern coast of the country, where stony coral tissue loss disease has become prevalent, to present new observations of stony coral tissue loss disease for the Dominican Republic and likely the Caribbean, and to discuss next steps and currently efforts being conducted to get a better understanding of this problem. Next one, please. So as far as the study area, I'm going to present results from 3 different locations in the northern coast: the east side of it, which is Samaná and Las Terrenas; the center, which is Sosúa, and the west, which is Cayo Arena and Punta Rusia. Next one, please. So if we look at the spatial distribution of this disease, it's clearly restricted to the northern side of the island, particularly in localities and sites located in the northeastern area. In the northwest area, the disease is in the endemic phase, whereas in the northeast area, particularly in Galeras and Samaná, the disease is in outbreak phase. So we have like differences in regions and localities nested within regions. Next one, please. So the host ranges of the disease is also varying across the scales. We have recorded over 10 coral species having this disease. And the SIMPER analysis shows that species that contribute the most with variation across locations and sites is Montastria cavernosa, Pseudodiploria stygosa, and Sargassum sierra. Next one, please. So what are the new observations that we have been making about this terrible condition? We have been observing recruits, baby corals affected by this condition. Over 9 species have been recorded so far. So Diploria stygosa, Dicocoenia stokesi, Orbicella, Eusmelia, and Sargassum sierra are the most affected ones. However, they are occurring in low prevalence. However, these results might be taken with careful, because we know that recruits are not as easy to record as adult coral. The problem, however, has been only observed in the outbreak zone, not in other areas in the Dominican Republic. The problem of stony coral tissue loss disease affecting recruits. Next one, please. Here we have some examples of some species that have been recorded having rapid loss disease for baby corals. Next one, please. Finally, we saw new observations of Oreaster reticulatus and Diadema antillarum feeding on slogging tissues of stony coral tissue loss disease colonies. We saw the problem in 3 coral species: Pseudodiploria stygosa, Pseudodiploria crigosa, and Colpophilia natans, for the particular case of Diadema antillarum. Oreaster was found in less than 5% of diseased corals and less than 1% of healthy corals in Samaná, and Diadema antillarum feeding on less than 1% of diseased corals in Fronton and Samaná. Next one, please. So here is some photos showing the behavior of the sea star feeding on slogging tissues. You can see the slogging tissue peeling off from the colony, and on top of that, you can see the Oreaster feeding, and on the right bottom side of the figures, you can see actually the ambulacra. When you remove the sea star from the diseased coral, you can see the ambulacra attached to the slogging tissue. So the meaning of this problem is not being addressed yet, but we are on our way to investigate whether this sea star could work as a potential vector for the disease. And I mean, with that, I would like to finish my presentation, and I am happy to take some questions from the audience. Yeah. Well, I don't know. I have a time.
Thank you, Aldo. No, we are holding questions to the end of the
Okay.
the research panel. So hold
So-
tight. Questions will be coming soon.
Okay.
So next, thank you, Aldo. Next, we're going to transition to Nuria Estrada Salvidar with the Biodiversity and Reef Conservation Laboratory. Nuria?
Okay, I'm going to talk about the ecological consequences of the stony coral tissue disease of the rapid spread in Cozumel. Next one, please. Okay, Cozumel is an important reef tourism site across the Caribbean. It has an estimated value of $300 million annually. The reefs in the southwestern part of Cozumel are among the most complex and best conserved reef systems in the Caribbean. They present high coral cover and high diversity. And although these reefs were among the last ones to be affected by this disease in the Mexican Caribbean, it spread very rapidly across the 20 kilometers of the southwestern Cozumel during the winter. Next one, please. So the means by which the disease arrived to this part of Cozumel remains uncertain, but we can talk about three hypotheses. The first one is that in September of 2018, we registered the disease in the east part of the island. So aided by the currents that run from south to north, it could have arrived to the southwestern part. The next one, please, is that recreational diving is a common activity across the Caribbean, and it is common for divers to travel among multiple locations within a single trip. So recreational divers could have brought the disease to the southwestern part of Cozumel from the mainland within their diving gear. And the third one is that the high influx of ships arriving to Cozumel may have served as a vector for the disease within the ballast water from the ships. Cozumel has 3 piers that receive more than 1,000 cruise ships a year, and one of the most important piers is located near the first site where the disease was detected. Next one, please. So based on this, our aim was to describe the spread of the stony coral tissue loss disease in Cozumel, to assess the effect of this disease in the benthic communities, and to evaluate whether the coral mortality was further exacerbated by the thermal stress events. Next one. A product of this, a product of this research, there is an article published within the special issue of the stony coral tissue disease in the Frontiers in Marine Science. Okay, yeah, thank you. Next one. We use 3 complementary approximations to assess the spread and impacts of the stony coral tissue disease outbreaks. The first one was prospective surveys were done between February and March of 2019. The second one, to evaluate the coral communities, we use Bell transects based on diagram methodology on July of 2018, July, August, October, and November of 2019. Also, we use the Bardurk method in January and April of 2019. And to assess the benthic communities, we use 6 30-meter point intersect transects for 6 sites in July of 2019, and they were repeated in July of 2019, and also 3 sites in April 2020. Next one. The stony coral tissue disease outbreak spread very rapidly along the southwestern part of Cozumel. In January of 2019, we found the disease only in one site in Paraíso. With a high disease, we took into account the disease and the recently dead colonies of the susceptible species. Next one. By the first week of February, we could see that the disease already spread along the southwestern part of Cozumel, but with a relative low disease prevalence. Next one. As the weeks pass, next one, please, we could see that the disease was incremented until July, where all the reefs had a high disease prevalence with a lot of death or disease colonies. Here we can see a common picture on the left one. We can see a lot of disease colonies of the susceptible species. And by July and November, what we saw were a lot of dead colonies that were covered with algae. Next one, please. Although in our transects, we did not register any colonies of the Dronera cylindrus, we did observe a lot of colonies that were severely affected by this disease. In January 2018, we could see this colony that was completely healthy. Next one. And 3 months later, it was almost completely dead. Next one, please. After the outbreak, we did so that the live coral cover decreased significantly. That went from 27% to 14%. And also the algae cover did increase after the outbreak. In the right panel, we can observe the coral dominated states that are in the blue box. And in the yellow box, we can see the algae dominated states. By 2018, we do not see a clear dominance of algae cover, but next one, please. In 2019, we can see that story change as there were a lot of sites with a higher proportion of algae than corals. Next one, please. And by 2020, we do not see any sites with a coral dominated states. Next one, please. As the common picture was to see a lot of susceptible coral colonies with overgrowth of algae, cyanobacteria, and other organisms. Next one, please. Between September and December of 2018, we observed an event that affected these corals. But regarding the coral mortality, we did not register that it increased due to the thermal stress. We could see that the coral cover before the outbreak was of 10%, and after the outbreak it decreased, but during the thermal stress event and after, the coral cover remained the same. Although during these months, there was a common picture, we could see colonies that were the photography on the top, that were bleached, but there were also disease and that have recent mortality. The next one were colonies that were bleached but with non-mortality. And the saddest one is colonies that had lesions of the stony coral tissue loss disease with other organisms overgrowing or other disease, but were also bleached. Next one, please.
2-minute warning, Nuria.
Yeah, I'm almost done. Although the devastating effects of this disease, there was a positive side of that. There was a collaboration between academics, the MPA managers, and the tourism sector to collaborate and to track the disease and raise awareness among the Cozumel community. They also did proactive activities such as colony treatment and rescue of susceptible species. The joint efforts that we saw during this outbreak that develop better management actions may help to improve the environmental conditions while enhancing the recovery of these ecosystems. Next one, please. And finally, I want to thank all the people who helped in the surveys and the National Commission to Protect the Natural Areas, as these data were part of our project. Thank you.
Excellent. Thank you so much, Nuria. And Nuria and Aldo, start keeping an eye on the chat box. Questions are coming in for you, but we will hold a discussion at the end of the research presentations. So next up, we have Claude Bouchon with Eco Recif Environment. Claude?
And Yolande, can you send the registration file?
Recording.
With content the sound?
No.
We can hear you. What? Sorry, what was the question?
We have posted a file with the sound included. Can you send it, send play?
No, we have to do it again.
Is there a video to play?
In the file that we sent to you, the sound is included.
Oh,
So
there's sound.
you can just send it.
No.
Oh, you want
No.
us to play sound on the video?
Yeah, the sound is on video. Just send the video.
Is it possible?
It's possible?
Oh, are you wanting us to play the presentation that was recorded? Is that?
Yeah.
Yes, possible.
Yes.
Oh, no,
No. Well,
we cannot do that. I don't think so.
no. Well,
Do you want?
We do it again.
Okay.
So we are Claude and Yolande Bouchon, and we would like to report about what happened in our region, in Guadeloupe. Next. So as you know, the coral disease were discovered by Antonius in 1973 in the Caribbean. And you know that now about 50 diseases are known in the world. And in the French West Indies, we have about 15 diseases. In Guadeloupe, the first observation occurred in 2020. In Florida, it was in 2014.
Yeah.
Yeah. And it appeared in Guadeloupe as in Florida after a coral bleaching event. In the Guadeloupe, the disease concerned more than 20 species. And we know about 60 in the Guadeloupe. Okay. And since 2020, the disease appeared. Qu'est-ce que j'ai dit? Can you put the before? Yes. So the disease was appeared in 2020, and since then all the reef of the island have been contaminated around all the islands. Next. So contrarily to previous disease, the SCTLD affects the largest number of species, but simultaneously. That's the difference with other disease. And the prevalence of the disease in Guadeloupe is very different depending on the species. Here you have the most affected species. I don't give the names. No? And the most resistant. That is important for the reefs. So the destruction of the tissues is very fast, as we observed, and can reach 2 or 3 centimeters per day. And the later outcome appears within a week or a month. Next. Here you can see the front line separating the healthy tissue from the dead skeleton. The part of the denuded skeleton, it's 3 centimeters large here, corresponds to about 20 hours before the settlement of algal turf on the skeleton. That gives a good idea of the rapidity of the necrosis. Yes. Next, please. Here you have 3 different stages of the disease. In diploria, on the upper side, the disease just began. Then the bleaching is complete, the disease is complete, and on the right, the skeleton, the coral is dead. And on the you have 3 other colonies of varieties, is that?
I say this.
And they were not touched by the disease. Next. Some example of the different species of coral that were affected in Guadeloupe, yes. As you know. Next.
Also affected fishes.
Yeah.
More affected species.
The most affected ones. Next, please. Next. So we think that the transmission is a vector mainly due to the coastal currents, and the expansion speed varies between 2 or 3 kilometers per day. When you see the disease in a site, you have 2 or 3 kilometers per day the day after, c'est ça? We couldn't link the seasonality to the disease and also to the coastal pollution. So as in Florida, it appears to be that the origin was bacterial. Then as you can see later, there will be some treatment for this disease. And in Guadeloupe, the applications of the treatment seems impossible because of the thousands of colonies involved and of the speed of the propagation. So Claude made a recent surveys with a Transect, and he observed the apparition of newly recruited colonies. And that brings some hope about the possible resilience of the coral communities in Guadeloupe. So there is some hope. Next.
Thank you.
Yolande, you have about 2 minutes.
2 minutes. Oh, but I finished. I answered the question with Claude after. Thank you for your attention.
Thank you so much, Claude and Yolande. Well done. Okay, next we're going to hand the screen over to Michael Studevan. Michael is with the Cooperative Institute for Marine and Atmospheric Studies at the University of Miami's Rosenstiel School of Marine and Atmospheric Science. Michael.
Thanks, Dana. I'm the coral disease lead at the CMIS coral program, as well as the NOAA Atlantic Oceanographic and Meteorological Laboratory. Our disease program has priorities in identifying disease transmission vectors. And so today I will be presenting on some of our recent work evaluating reef sediments as a stony coral tissue loss disease reservoir. Next, please. So an increasing number of studies and hydrodynamic models are now suggesting that water is the primary transmission vector for the disease, as seen in its rapid spread throughout the entirety of Florida's coral reef tract. Next. And a recent study led by Cynthia Becker and the USVI found that there were 25 different microbial indicator taxa found in diseased corals and in their surrounding seawater. Next. Additionally, another one of our researchers here at AOML, Stephanie Rosales, found 2 higher order microbial taxa to be associated with diseased corals and the surrounding sediments. And so she hypothesized that sediments are likely playing a role in transmission and persistence of the disease in Florida. Next. And so we set out to test whether reef sediments can serve as a reservoir or vector at local scales using an ex situ transmission assay. Next. We started this experiment by first collecting 3 entire diseased colonies of Montesquieu cavernosa and 35 liters of reef sediments from a site in Broward County in southeastern Florida. Next, please. Following autoclaving the sediments, we then incubated them for 2 weeks in 2 separate aquaria with and without those diseased colonies. So in the top infographic there, we have a healthy sediment. In the bottom, we have a disease inoculated sediment. Following that 2-week incubation, we homogenized each of those sediments independently of one another, and then we dispensed 150 grams of sediment into each of 4 different treatment jars with a water flush to minimize chances of waterborne transmission. So in the top, we have individual sediment transmission that I will describe in just a moment. We have our healthy sediment. We have a disease contact treatment that has a bed of healthy sediment and then a fragment of the disease donor colony to maintaining direct contact between the donor and a healthy fragment. And then we have a batch disease sediment using that disease inoculated sediment. Next. As the corals in our disease contact treatment became infected and were removed from the experiment, we then used each of those individually inoculated sediment jars for our individual sediment transmission. So we're comparing 2 different modes of sediment inoculation here, being the batch transmission that has entire colonies of Montesquieu cavernosa, and then the individual sediments that are small little fragments, about 5 square centimeters of diseased corals. Throughout the experiment, we made daily observations and photos to quantify the time to transmission, as well as taking some downstream samples that I will describe in just a moment. Next. Jumping right into the methods here, or into the results here, this plot is showing the average time to transmission for Orbicella fabulata. So we have our time to transmission in days on the y-axis, and our sediment treatments on the X. So in the light tan, we have our disease contact. The dark green, we have batch disease sediment, and then the lighter green is an individual disease sediment. And what we noticed is that our batch disease sediment and our disease contact treatments were about the same in terms of the onset of disease, where disease occurred about 7 days after initial exposure. However, what was really striking is that the individual disease sediment treatment, all of the corals that became infected manifested disease lesions within 24 hours, which is very surprising. It's a very rapid onset of disease. Now, in the bottom left panel here, we have the transmission rate, or the proportion of diseased corals within the treatment. And as expected, our disease contact treatment had much higher rates of transmission, around 90%. And both sediment treatments were lower, coming in at 33%. Next. For Montesquieu cavernosa, we did observe some species-specific differences, where the onset of disease in the disease contact in the batch sediment treatments were longer. This has been shown in previous transmission experiments, where it takes longer for Montesquieu cavernosa to develop visible lesions, took on the order of about 14 days or 2 weeks. But once again, we saw the really striking difference with the individual sediment treatment, where we had disease transmission occurring within 24 hours. Now, we also did see a species difference in the transmission rate in the bottom right panel, where we had 100% of our disease contact corals get infected, with between 67 and 83% for the 2 different sediment transmission treatments. So this would appear that there is species-specific variability in terms of transmission rate, as other experiments have also shown. Next. Now, these coral samples were sent off for a histological examination, and we did confirm presence of disease symptoms in all of the disease treatments. But what I want to point out in this photo panel here is that the lesions manifested very differently between the 2 species. Where in Orbicella fabulata, we see the characteristic white lesion that progresses rapidly across the entire coral fragment. But in Montesquieu cavernosa, the symptoms were often more subtle. In the top right photo, we had evidence of excess mucus production around the margins of the coral. In the center right photo, we also had evidence of tissue necrosis, those brownish and white patches that are occurring on the margins. And then finally, we had tissue retraction in the bottom right photo, where the tissue was pulling back from the margin of the coral. And generally, the initial disease symptoms manifested themselves on the sides or bottom of the fragment, where there was closest contact to the inoculated sediments. Next, please.
1 minute warning, Michael.
Thank you. We also sent off sediment samples for microbial community profiling with 16S, and we identified that there were 33 differentially abundant genera among sample types. So instead of comparing among sediment treatments in this case, we compared them between coral-associated fates. So we had control corals that were never exposed to disease, no active infection corals that were exposed to disease but didn't demonstrate infection, and then corals that were exposed to disease and saw tissue loss. In addition to the 33, we had 16 of those genera that were more abundant in tissue loss associated samples. Next. We also found that 15 of the 25 microbial indicator taxa identified by Cynthia Becker were present in our sediments, including 14 members of Vibrio that were more abundant in our tissue loss samples. Next. So we found that reef sediments can serve as a SCTLD reservoir, which has direct managerial implications for mitigation of disease spread in coastal zones, including the Port Everglades expansion set to occur in the next couple of years. Next. So we are wondering whether transmission risk can be reduced through treatment of inoculated sediments or other actions, and how, say, dredge barges handle sediments before disposal. Next. There are likely additional factors that affect the transmission rate, including how long sediments can remain infectious and whether there are species-specific interactions. Next, I'll wrap up very quickly. And we found that there are indicator microbes that exist in these reef sediments. And what we don't know right now is whether these represent opportunistic growth related to increased organic matter, or if they're representing potential pathogens found in the sediments. And since I am out of time, I will thank you for your attention, and I'll be happy to take questions in the Q&A session later. Thanks, everyone.
Thank you so much, Michael. And just reminding everyone that the recordings of all of these presentations and 10-minute versions are available on Whova. So up next, we're going to hear from James Evans. James is with the U.S. Geological Survey in St. Petersburg, Florida. James.
Yeah, thanks so much. I'm really excited to share some of our ongoing research into potential links between SCTLD and biofilms. Next slide, please. Thank you. Yeah, so we've already heard a lot of really great background information about SCTLD, so I'm just going to highlight a few points that are relevant to our ongoing research here. Specifically, of course, we don't know the causative agent of the disease, but we do know that the disease is transmissible through the water column, and there's the potential that bacteria are involved in disease progression because antibiotics have been shown to be successful in treating disease lesions, and also molecular investigations have found specific bacterial taxa that are consistently enriched in disease lesions. Next slide. Great, thanks. So within the Florida Reef Tract, progression of SCTLD has been consistent with movement via natural water currents, but throughout the Caribbean, outbreaks have been a lot more sporadic and have often occurred near major ports. And so that suggests that ships are potentially involved in the spread of the disease. And biofouled ship hulls have long been recognized as a vector for the introduction of non-native species into new regions, but what we don't know is whether maybe biofilms that form on ship hulls or inside of ballast tanks could potentially contribute to the spread of the disease into these new areas. Next slide. So since we know that SCTLD is a waterborne disease, that means that whatever the causative agent is, then infected corals must shed it into the surrounding water. But to get at this idea of biofilm transmission, what we need to determine is whether those shed microbes are even capable of forming biofilms. If they are, do we see differences within those biofilm microbial communities, such as differences in the donor corals or the corals that are contributing or shedding those microbes? Do we see differences in like the coral species reflected in those biofilm microbial communities? And finally, and most importantly, do we see an SCTLD signal? Next slide. So to tackle this, these three questions, we first filled 15 18-liter mesocosms with seawater that had been UV treated and 0.2 micron filtered, and that was just to reduce the starting microbial load. Each one of those mesocosms then received a coral colony, and we had 4 healthy colonies. These were colonies that were collected from the reef and then held in quarantine for 1 or 2 years, just to make sure that they were truly healthy individuals. And then we had 10 diseased colonies, and these were colonies that were fresh off the reef and exhibiting visual symptoms of SCTLD. We also had a seawater control that didn't receive a coral, and all of the corals that we used were the same species between the healthy and the diseased treatments. Each of the mesocosms also received a cleaned and autoclaved stainless steel plate to serve as a proxy for ballast tanks and ship hulls. And those were set in petri dishes to allow for water contact on all sides. And then we incubated those mesocosms for 3 days. Then we scraped the plates with razor blades, rinsed them with a sterile buffer, and we extracted DNA. Next slide, please. So then we performed next generation sequencing of the V4 region of the 16S rRNA gene, basically targeting the bacterial and the archaeal communities using those universal bacterial and archaeal primers on an Illumina-MiSeq platform. We got about 30,000 sequences per sample back, excluding our negative controls. And those were processed within Shine 2 and assigned to 100% amplicon sequence variants, ASVs. Basically, when I say ASV, you can think of a bacterial or archaeal species. Next slide, please. So now we're looking at some results. So this is an Emperor PCOA plot, principal coordinates analysis plot. And basically every colored point that you see here represents the entire bacterial archaeal community for a single sample, in this case, a single mesocosm. And the axes indicate the percent variation represented by that axis. So that the takeaway is the closer that 2 points are to one another on this plot, the more similar their overall microbial community structure. So here we are grouped by the health status of those donor corals. So the health status of the mesocosm, diseased is red, healthy is blue, and that lone yellow dot is our seawater control. And we can kind of see right away that there are some differences showing there between our diseased mesocosms and our healthy mesocosms. And over on the left, we can see, based on our permutational multivariate analysis of variance, PermaNova, that those differences were in fact significant. There was no significant difference detected between the diseased or the healthy mesocosms and that seawater control, but that's probably an artifact of our sample size of 1. Something else that's important to note here is that there are some other potentially contributing factors that are difficult to pick apart. So you might remember I said all of our healthy corals were from aquaria, all of the diseased corals were fresh off the reef. So if we were to plot here aquarium versus reef, we would get an identical plot to this one. But next slide. Suggesting in particular that the disease status of our donor coral is contributing more directly to those microbial communities than maybe other donor coral characteristics was the detection of numerous SCTLD associated bacteria within almost exclusively the diseased mesocosm. So Becker et al., as we just heard, identified these 25 bioindicator, SCTLD bioindicator ASVs. And those 19 of those were detected in our biofilm data set, and those are shown in purple. And the identity within our microbial communities is shown in gray there. And Becker, these were 100% sequence matches. And Becker et al. Also identified 4 of these ASVs, which are shown in bold in that purple column, as being 100% identical to the overlapping region for ASVs that were also enriched in SCTLD lesions in 2 other studies. Interestingly, on the next slide, if we expand this analysis to just look at ASVs that were at least in 1 lesion in these other studies, we see an even greater extent of overlap. It's of course unknown whether any of these ASVs are actually implicated in SCTLD, but the extent of the overlap that we see here, which spans different regions, years, coral species, now source material, certainly suggests that they warrant additional investigation. This table also shows us the total number of reads for each ASV detected in each one of our mesocosm types, with the diseased in green, healthy blue, the seawater control is yellow, and our positive and negative controls are orange. And we can see that the vast majority of these SCTLD bioindicators were exclusively found in our diseased mesocosms. None of them were found in all of our diseased mesocosms, but 2 of them, which is what those black boxes indicate, were found in 70% of our diseased mesocosms. The far right column there just shows the lowest taxonomic ID of each one of those ASVs, with a notable number of Vibrios represented, which is a genus that includes known coral pathogens, as well as some other taxa that have been indicated as potentially of interest in SCTLD. Next slide.
One minute warning, James.
Thank you. Yeah, so just our quick conclusions here. We did find that the bacteria and archaea that are shed by corals are capable of forming biofilms, and that we can detect differences within the between the donor corals within those biofilm microbial communities. We also found a potential SCTLD signal within those biofilm microbial communities. And so the next question, of course, is whether biofilms are actually capable of transmitting the disease. So we'll be tackling that question with a transmission study in the near future. So stay tuned for that. On the next slide there, I just want to thank the many, many people that made this work possible. I'll wrap it up and I'll take questions in a moment, but thank you very much for your time.
Excellent. Thank you, James. Everyone, keep writing down your questions and either type them in the chat or be ready to type them in soon. Next, we're going to hear from Catherine Eaton with Moat Marine Laboratory. Catherine?
Hi, everybody. Thank you so much for tuning in. I usually go by Katie, actually, but today I'm going to talk to you about a project that we did in our lab. I work for Erin Muller at Moat Marine Lab, and we looked at the microbiomes of apparently healthy and stony coral tissue loss disease-affected Montassia cavernosa and Orbicella fabulata. And these samples were collected from a transmission study that we completed in our lab. Next slide, please. So just some quick background information on stony coral tissue loss disease. As it's already been shown, it has spread to other areas of the Caribbean, as well as the entirety of Florida's coral reef. Over 20 of major coral reef building species are susceptible, with 5 of those listed under the US Endangered Species Act. And then the uniqueness with this disease is the high prevalence it has in some species, as well as the high rates of mortality. Next slide, please. So stony coral tissue loss disease, the pathogen is still unknown, but it likely has a bacterial component. Some other studies have found that stony coral tissue loss disease lesions respond to antibiotic paste, which is essentially amoxicillin mixed with a base 2B paste that's applied directly to the lesions. And we see relatively high success rates with these treatments. A couple other studies have also used a 16S sequencing approach to characterize the stony coral tissue loss disease microbiome and identify significant ASVs. These 2 studies here were some of the first research done on stony coral tissue loss disease microbiome, but we're still unsure whether the identified bacteria are primary pathogens of the disease or secondary opportunistic infections. Next slide, please. So the work that we're doing here contributes to the consortium of stony coral tissue loss disease microbiome data that we have. So the objectives were to execute 2 different disease transmission studies using apparently healthy Montassia cavernosa and Orbicella fabulata with diseased Montassia cavernosa. This was conducted in our wet lab, and we collected samples to identify key differences in the bacterial community of apparently healthy and diseased corals. So we had stony coral tissue loss disease exposed corals in tanks, and then we also had apparently healthy control tanks as well. We also wanted to examine how the bacterial community changed over time within a fragment. So we took samples at a variety of different progression points as the disease progressed within a fragment during our experiment. Next slide, please. And this just gives you an overview of our methodology. So we had 2 different transmission studies with disease tanks and apparently healthy control tanks. For sample collection, we scraped with a star razor blade and samples were flash frozen. Each time a disease sample was collected, paired apparently healthy control sample was also collected. Samples were sequenced on the MiSeq platform using the 16S rRNA V4 region using these primers, and then data was processed using the Data2 pipeline and Silva and R. Next slide, please. And to give you an idea of the amount of samples that were collected, we had anywhere between 7 and up to 30 disease samples with paired control samples as well. Next slide, please. And jumping right into the results. So looking at the first experiment, exposing Montassia cavernosa healthy to diseased Montassia cavernosa. We did find that rhodobacters, rhizobiales, desulfovibrio, and Clostridia were found at higher relative abundances within disease samples. These results are from an ANCOM analysis, and we did do our analysis at the ASV level, but for this, we just combine them for visualization purposes to look at the significant taxa. We also found that the relative abundance of Clostridia and desulfovibrio increased as the disease progressed through Montassia cavernosa fragments. And rhodobacters and rhizobiales were not present in any of the apparently healthy control fragments. Next slide. And now looking at the fabulata exposure, the analysis was done the same way as the first experiment. And we did find that rhodobacters, rhizobiales, desulfovibrio, and Clostridia, again, were found at higher relative abundances in our disease samples, and the relative abundance of Clostridia increased as the disease progressed through the fragment. Next slide. So some main conclusions we can draw from our study. The disease-associated taxa that we found in our study has been found in other microbiome studies. Again, I still need to compare the significant or specific ASVs with ASVs from other studies as well, but we are seeing the the taxa across multiple studies. And it's important to note that this taxa is associated with other coral diseases as well. So it's possible that there's potential co-infections or secondary infections. For example, we saw high levels of disulfovibrio in our cavernosa experiment. So it's possible that black band was a potential secondary infection. These disease-associated taxa are also found to be pathogenic to humans, animals, and or plants as well. Next slide, please.
Oh, I was going to give you a 2-minute warning, but it looks like you're wrapping up.
Yeah, wrapping up now. Thank you. So some future directions we want to go with this study. We want to look at our specific ASVs and compare them to other studies as well and identify consistencies. We're also planning to work with collaborators to study the function of these bacterial taxa using metagenomics. There's also potential to isolate these bacteria in culture to attempt inoculation studies. And we'd also like to use this information to further develop mitigation strategies. And with that, I just want to acknowledge the State Wildlife Grants Program for funding for this and the Sanctuary for providing permits and my interns and fellow colleagues as well. And I'll be happy to answer any questions at the end. Thanks.
Thank you, Katie. Thank you very much. And I just want to comment that I love all the connections that I see happening in the chat and our presenters making connections between their research, talking about coordinating. That's great to see. Our final presentation in the research component of this morning's session is our student presenter, Caroline Dennison. And Caroline is competing against other student presenters in the GCFI conference here. So Caroline, take it away.
Alrighty, thank you very much. And thank you guys for coming and tuning in today. Today I'm going to be discussing the results of an experiment conducted in 2020 investigating the role algal symbionts in the genus Breviolum play in SCTLD susceptibility. And as you can see, there's quite a few folks who have collaborated on this project to look at things like histology, gene expression, and microbial dynamics. But the primary focus of this talk is going to be on the experiment itself. Next slide. So the goal of this project was to really look into the role of algal symbionts in SCTLD susceptibility. And this interest was based on some of the key observations that were made early on, the first being the tissue pathology. So histology notes that the first cell layer affected is the gastrodermis. And this is the cell layer that houses the algal symbionts. Next, I want to highlight the susceptibility hierarchy, which notes that not all species are equally susceptible. And this can be based on the host or looked through the lens of the holobiont, which includes the algal symbionts. And finally, overall, just looking at disease dynamics, what we typically see is that the disease incidence declines during the summer months when bleaching occurs. Which could be the result of the loss of associated algal symbionts. And so as I've kind of alluded to here, at the center of all of this is the interactions, at the center of all the interactions and observations are really the coral host algal associations. So ultimately the goal here was to look at algal symbionts as a driver of SCTLD susceptibility and resistance. And to test this hypothesis, we collected colonies from the dry tortugas from 5 different species. And at this time, we collected in January of 2020, the disease was not present in the dry tortugas. And from these colonies, we generated replicate cores, which were then used in some lab experiments where we we did bleaching controlled and recovery. And overall, the goal was to compare cores with Breviolum to cores manipulated to have duristinium or very few symbionts. And then following these manipulations, we exposed cores to a disease bath. Overall, we saw a clear effect of algal symbionts. Cores associated with duristinium and cladocopium were significantly less likely to present with SCTLD when compared to those with Breviolum or bleached. They don't appear to be immune though. And using a mixed effects model, which you can see in panel B, in addition to a species susceptibility hierarchy, there also looks to be a symbiodiniaceae susceptibility hierarchy, where Breviolum is the most susceptible, followed by cladocopium and duristinium, which are about equally susceptible. And And then based on our field observations, Symbiodinium, which does not appear to be susceptible at all. On a grander scale, the results of this experiment can help explain some of the field observations that we see in the intermediately susceptible species. In particular, looking and comparing the spatiotemporal algal mosaics in some colonies and relating that to the actual disease. We see that some parts of the colonies may become diseased while others do not. This also has implications for restoration, since we can predict what environments select for Durstinium. And once we know that, we can select colonies to use for fragmentation and grow out and ultimately use them to restore heavily impacted reefs. Next slide. So this data that I was working off of is looking and assuming that the cores are only associated with a single algal genera. However, we know this is not the case based on our quantitative PCR data. So instead of ranking as a factor, symbionts as a factor, we also rank them as a continuous trait based on this qPCR data. And from this, we developed a Cox proportional hazards model to calculate relative risk based on the proportion of algal symbionts. Next slide, please. And from this, we can see that the relative risk will actually fall below 1, indicating it is less likely to become infected when there's about 20% Durstinium in coral tissues. Next. And this, like I said, means that there's less of a chance of infection. On the other hand, I think we are 3 next away now. Next, maybe. Is it working? Another one. One more. Okay, sorry. A colony containing around 75% Breviolum, the relative risk will actually climb above 1, indicating that it is becoming more likely to become infected with SCTLD. Next slide. And when we look at this side by side, we still note that Durstinium does not appear to be immune. However, there is some uncertainty here that really cannot be accounted for, since the data is working off of a single sample point, and it can't account for day-to-day shuffling that may occur within coral tissues. So, one of the things that we know is that even minor shuffling may have a pretty big effect on a coral's susceptibility. Nonetheless, associating exclusively with Breviolum carries a risk 2.5 times greater than that of exclusively hosting Durstinium. Next slide. And so I really I could talk about this all day, but I really want to get into some of the conclusions that we came up with here. First off, there really appears to be a clear effect of algal symbionts on SCTLD susceptibility, and this really begins to elucidate some of our field observations that we don't fully understand yet. However, the major takeaway from this project was that in addition to a species susceptibility hierarchy, we also see a Symbiodiniaceae susceptibility hierarchy. And we propose that Breviolum is much more susceptible than Cladocopium and Durstinium, which appear to be about equally susceptible. But all of these are much more susceptible than Symbiodinium. I also just want to take a moment to note that the results of this experiment kind of point out that Cladocopium and Durstinium dominated hosts may not be immune. But I think it's important to note that a 2.5 times increase in a relative risk in a disease bath may have a may be very significant when translated to the field. This was an experiment that was conducted over 60 days. And during these 60 days, these cores were exposed to constant disease influx. They never went a day without disease water. So that may have implications for their bacterial the bacterial side of this. I also just want to note that the analyses are still ongoing and the collaborators are still chugging away at this data. We're currently using the samples to look into whether algal symbionts are the direct target of SCTLD or if they affect the susceptibility indirectly by modifying the host immune response. We also have a subset of data that's being used for 16S and 18S analyses as well. And next slide. With that, I would just like to acknowledge everyone who has helped along the way. We conducted the the crews to collect these colonies, and there were a lot of folks that were able to help out with that, including getting the permits. So huge shout out to everyone who helped with permitting as well as just the collections. I also want to point out that Karen Neely has been extremely helpful in all of our projects in providing disease coral colonies as source disease. So a huge shout out to Karen. We have some ongoing work right now, and she's still extremely, she's providing everything that we need. And I would like to thank FDEP as well as the NSF for funding this research. Next slide. And with that, I'm happy to field questions now. I'm also happy to field questions via email that you can see.
Thanks, Caroline. We're actually going to hand over to Emma, who's going to facilitate some Q&A and discussion with everyone. If you haven't already, please go ahead and enter any questions you have for any of our speakers or for all of our speakers in the chat. Emma, handing over to you. And I just want to say thanks to all of the presenters for staying on time. Much appreciated.
Yeah, good timing. Okay, so we've got a number of questions that have come through for Aldo, some comments for Nuria, Cloud, and Yolande, for James, and also for Caroline. I'm looking at the Q&A and the chat, and also at the WOVA app. Now, Aldo, questions have come in for you regarding opportunistic feeding by fireworms and snails. Have you noticed that? And is it really opportunistic feeding by diadema and Oreaster? From Alize and from Judy.
Thank you, Emma. Yes, indeed, there is a lot of opportunistic feeding that you can see in areas where stony coral tissue loss disease becomes, in outbreak phase. We have seen fireworms. We have seen the diadema, we have seen the Oreaster, we have seen the butterfly fish, and many others. However, the significance of this is not yet clear. For other rapid loss disease, such as white plague, we know that some of these animals might become like vectors, but that is not clear for the stony coral tissue loss disease as far as I know. And I see a question from Judy. I think that's a great point, Judy. We don't know yet if Oreaster behavior is actually a problem. It might be the case, as you suggest, that when the starfish eats the tissue, it might destroy the pathogens and could actually work as as a factor that heals the coral. But also it could be the case that the sea star could be touching, not necessarily digesting, but touching the diseased corals and then move into a healthy coral and become like a vector. We don't know that yet, but that's a great point.
Okay. Any advice from you, Aldo, or anyone else on collecting examples of some of these opportunistic feeders?
Yeah, getting the permits here is kind of complicated. What we have done so far is to collect sample tissues to start to know if we are dealing with the same kind of pathology across locations. What we know now is that coral seems to have stony coral tissue loss disease, but could be another type of rapid loss disease such as white plague. So we are now in the phase of knowing what we have in there, and potentially we will try to conduct some experiments, but we didn't have the facilities to set up experiment to test specific hypothesis on behavior of Oreaster. Let's see what happens. I just came back last week from the place. I saw Oreaster, and sadly there is no more colonies infected because all of them are dead. But we'll see what happens.
Okay, thank you. Muchas gracias, Aldo. There is a question from Alfonso Aguilar Pereira. Perhaps you can answer that in writing in the Q&A box that's just come in. I want to move on to some perhaps some easy questions for Claude and Yolanda Bouchon. Judy Lang is asking, where did the disease first occur in Guadeloupe? And does that give any clues about how the disease reached Guadeloupe?
We don't know exactly because we were in a period of COVID and it was forbidden to use boat and to go at sea. So we discovered by chance in May in Guadeloupe, and the disease was well installed at this time. In January, there is no disease, so it appeared in Guadeloupe between January and May, and we don't know exactly where and exactly when, because it was forbidden to use boat and dive around Guadeloupe because of COVID.
Okay. Patricia Kramer is asking, in Guadeloupe, how deep have you observed stony coral tissue loss disease? How deep?
How deep?
At what depth?
Oh, until 40 meter deep.
Okay. All right. Thank you very much.
Sorry, we didn't dive deeper, so I don't know if it exists deeper, but I have no report for friend diving, making deep diving about the disease deeper, but I don't. I think it's possible it exists deeper.
It's deeper.
Okay. There are some comments in the question and answer section. There are some interesting comments about the maximum depth observed as more than 130 feet in the Turks and Caicos Islands, and as shallow as 1 foot deep. And there's also some interesting comments provoked by Ellery Lennon regarding whether there are any areas of reef that are safe from the disease, that have been resilient to the disease. Nuria has answered there, so you might like to take a look at some of the areas reported not to have stony coral tissue loss disease. That's something that we're also very keen to track. Is where
in Guadeloupe, in Guadeloupe there is no safe zone. All the reef around the island are touched by the disease.
Okay. All right. Thank you very much.
And deeply touched.
Okay. Thank you very much, Claude. We've got some good exchanges going on. Oh, just one other question. Sorry, Claude and Yolanda. Yep. From a question about what are you defining coastal pollution as in relation to the disease?
Okay, we have an area in Guadeloupe where submitted to more or less heavy coastal pollution, and we have a clean area. And the disease, there is no difference in the expansion and the number of colony touched by the disease in polluted area or out polluted area. So we don't think that the pollution have an effect of the disease. The cleanest area around Guadeloupe are deeply touched by the disease. And in the same way, we didn't find any seasonality, apparent seasonality in the expansion of the disease.
Okay, thank you very much. Let me go to Nuria. There's a question from Alfonso Aguilar Pereira.
Sorry,
asking, yeah.
We are obliged to leave you. Good
okay.
continuation.
Okay, thank you, Claude.
Okay, thank you,
Merci.
Claude and Roland. Merci.
Nuria, a question for you from Alfonso Aguilar Pereira asking, what is the most compelling evidence that recreational divers are vectors of stony coral tissue loss disease?
And this in Spanish?
Certainly.
Okay. Hola, Alfonso. Pues hay varios estudios de la NOAA donde indican que los equipos mal lavados podrían ser vectores de patógeno que sea. Para nuestra investigación monitoreamos los arrecifes que están al este de Cozumel y en septiembre de 2018 vimos que tenían esta enfermedad. Como sabes, en Cozumel los arrecifes mejor desarrollados están al sur, entonces es donde nos concentramos, igual donde está el área protegida. Pero si tú tienes la información, pues me encantaría platicar sobre esos arrecifes.
Okay, gracias, Nuria. James, we have a question for you from Judy Lang, saying that she's heard that viruses can also occur in biofilms. Is there any chance of also looking for evidence of viruses in your sampled materials?
Yeah, thank you. Thanks. I think that there is a chance of that. I think that's very interesting, and I think some of the ongoing work is suggesting that the viruses are potentially implicated in the disease. We do still have those DNA extracts. The DNA extraction kit that we utilized was a power biofilm kit, so optimized for biofilm DNA extractions. I'm not really sure exactly how well that targets viruses. So that would be something that I would definitely need to look into a little bit further before we would actually tackle that question. But we do have filters related to transmission of the disease and part of a different project that are specifically targeting the viral size fraction, that if we were interested in looking further at potentially viruses that are associated with SCTLD diseased corals, we do have that information available. We just haven't actually extracted the DNA from those yet. So
if I could just jump in for a moment. So the particular virus that's now suspected to be a primary pathogen for this disease, that was first identified by Terry Work, the paper of that study is actually now available through Frontiers. So for anyone that's interested, it's part of the special research topic on SCTLD. But anyways, the virus that they identified through microscopy methods is likely an RNA virus. And so most of the existing microbial work that's been done would not really capture the viral component related to the disease. And so I think one thing that would need to be done is all the groups that are doing microbial work would need to go back to those original samples and work them up for metatranscriptomics rather than metagenomic analyses. So hopefully everybody's got some good sample archives remaining, because I think we need to start looking at the viruses.
Do you have any advice for people around the region in terms of keeping samples like that?
Right. So our group has had really good luck with a relatively new preservative. It's called Zymo RNA and DNA Shield. It is a room temperature stable preservative, which is probably the best part about it. A lot of the previous preservatives that have been used in the field require refrigeration or freezing. So this is room temperature stable, and it is completely safe to ship or to fly with, which is another big component. So I would definitely suggest using Zymo Shield. And all you have to do is take maybe a 5 ml tube, take a scoop of sediment, top it up with Zymo Shield, and then that can be frozen or kept at room temperature for up to a month, I believe. And we've gotten very consistent yields from it. So I would say that's the best option.
Okay. Can I ask you to pop the name in the chat
in fact,
for everyone?
I will do one better, and I will find the link for it.
Okay.
I actually just posted Terry Works.
Oh, there you go.
Oh, thanks, Dana. I came across it yesterday. It's very, very exciting to see that was accepted. And I will also mention on the Zymo front, they also now make pre-filled feed tubes, so they're pretty much ready to go for extractions. You can buy them in kits of tubes, so anyone can buy those and take them directly in the field. Take a scoop of sediment, pop it in, and you're good to go. So it makes it just about as easy as possible. I'll put some links in the chat.
All right, thank you both. James, there's another quite technical question from Aldo. Aldo, I wonder if you might copy that question directly to James for me on some statistical analysis questions there. I'd like to just move on to a number of questions for Caroline, which is important as our student. Caroline, there's some questions coming up in the chat, and there's also some questions for you in the WOVA app. So you might have a look at those. If we don't get to them, we've got a few minutes left here. The questions, there's some interesting questions here. Aldo is saying, is there any variation in the relative proportion of these symbionts within the same colony? And if so, does that mean that some areas of colony are more or less susceptible to infection?
Yeah, that's a really great question. So within a colony of, say, Orbicella fovulata, which is an intermediately susceptible species, you can have different proportions of algal symbionts across the colony. You can also have different algal symbionts in certain parts of the colony, which is why I mentioned the mosaics that exist and how that is related to the disease dynamics in Orbicella fovulata. We do have an ongoing project with Brian Walker, is currently looking into this mosaics and relating it to the actual disease incidents that we see in the field. So yes, those types of relationships do exist, and we are looking into them. I just have anecdotal evidence right this second, since I don't have the whole picture for the 1 year mosaics. Yeah.
Okay. All right. There's another question for you from Alfonso Aguilar Pereira. Is there a relation between symbiont genera in the coral and resistance to stony coral tissue loss disease? Which one would die first, the symbiont or the coral polyp?
Yeah, so I was actually responding to this in the chat.
Okay.
Right now, to be honest, we don't know the answer to the latter question. So what would die first, the symbiont or the coral polyp? Anecdotally, what we do know is that the coral tissue just appears to die. However, we are running a formal experiment comparing the survivorship as well as the disease incidence in coral hosts, so algal symbionts within a coral host, as well as cultured algal symbionts. And I actually, right before I hopped on this meeting, we were getting preliminary results. And I do not believe I can share those yet, just because it's the first round, but we are seeing some interesting dynamics there.
Okay. Now, over on the app, you've got a question being asked by Gretchen Goodbody Gringley, asking about the uptake. Let's see. This is interesting implications. Could inoculation with less susceptible symbiont species in the lab be a possible mechanism to restore affected populations of susceptible species?
Another phenomenal point. We are doing this. So we actually have coral juveniles, so colonies that spawned, I believe, beginning in May of this year. We've crossed them, we've used cryopreservation, we've done normal crosses, and we've had fertilization, we had larvae, the larvae settled, and we've actually inoculated the larvae with the cultured symbionts that we're using in the other experiments. And those, we actually have a pretty major experiment going on now that includes adult corals, these juvenile corals who have been inoculated with cultured symbionts, as well as Indo-Pacific and Pacific species, looking at the disease susceptibility. So it's a pretty major experiment we have going on, but we are doing that. And that is one of the techniques that we're attempting to use, since the coral juveniles appear to be more likely to keep their symbionts while they go out into the field.
Okay, all right, terrific. Perhaps you can go over to the app and finish and also type in a couple of answers to the other questions waiting for you.
Yes.
All right, before we finish up on the question section here, there are some comments from Matteo asking about citizen science involving fishers and tour operators in monitoring for the disease. Matteo, I can see that Nuria is responding to you, but we also, in the poster session for this, for stony coral tissue loss disease, there's a couple of posters that speak about citizen science and the involvement of local community members in monitoring work, in treatment work related to the coral disease. So you might go and check out some of that work. And Alize, who's on the screen, is also a good source of information on that. So I think with that, Dana, Caroline, do you have any other questions that you'd like to pose?
None for me, Caroline. If you don't have any, we'll transition to our second panel. Okay, thank you so much for the great questions and discussion and presentations, everyone. Moving forward to our second panel of the session, we're going to focus now on the action in the field to combat stony coral tissue loss disease. We're going to head first to the Turks and Caicos Islands and hear from the famous Alize Zimmerman about all of her hard work and their amazing efforts. Alize has been at the forefront of and so good about such a great spokesperson for all of the great effort going on in the Turks and Caicos to combat the disease. So Alize, handing over to you.
Hi, everyone. Thank you, Dana. My name is Alize. I'm the Executive Director of the Turks and Caicos Reef Fund. We are a nonprofit NGO established here in 2010 to help preserve and protect the environment of the TCI. Since the arrival of SCTLD in our waters in 2019, we have been on the front lines of the fight against stony coral tissue loss disease and try to help as much as possible in the regional effort as well. Next slide, please. Although I am officially an employee now, we are reliant almost entirely on volunteers, fundraising, and private donations. And this little case study here is just to show you a little bit of what we've been able to accomplish in the last 2.5 years of dealing with SCTLD and against a lot of odds as well. So next slide, please. SCTLD was first observed in the TCI by members of the School for Field Studies in South Caicos in January of 2019. And it wasn't observed again until a few months later on the opposite side of the archipelago in April at the southwest point of West Caicos. It then made its way following the predominant local currents around the Caicos Islands with a pause that I'll tell you about in a moment. And then it wasn't until December of 2019 where it made the jump against the 44-kilometer wide Turks Passage and was found on the Turks Bank. It wasn't until March of 2021, so this year, that we had our final observation confirming presence on all the 4 reefs of the Turks and Caicos, which was at East Caicos, which is an uninhabited island. We don't have a guarantee of when it first arrived there, having not been able to monitor that area, but I did just come back from an amazing week of research out there. And although there is presence, it's still a jewel of a reef with incredible genetic diversity that is still found on those islands. Next. So I mentioned that we had a little break, and so to note is that in September of 2019, as others have mentioned, we did see a slowing down, in fact, a stopping, both the colony and reef tract level of the spread of SCTLD when we had a nationwide bleaching event. And it wasn't until September of 2020, a year later, that we saw a reemergence of the disease on the Grace Bay eastwards through the Caicos Keys. And since then, we've been back in full outbreak. But if we fast forward to 2021, so on our next slide, please, based on the field trials, test trials that we did, the Turks and Caicos Islands government awarded TCRF with an treatment expansion scientific research permit, which allowed us to go forward and treat more priority colonies along the reefs with more opportunistic monitoring. This summer, we began conversations with different organizations to try and get the knowledge we need to build the Turks and Caicos Islands' first land-based facility for coral research and propagation. And in October, we had our very first rum-powered research expedition, which was what allowed us to go out to East Caicos and collect some incredible data and increase our education and outreach facilities. Next slide, please. This slide here is really more of an just a visual representation of how widespread SCTLD is affecting our reefs, and it's alphabetical rather than susceptibility. You can see we do have some entire taxa included here, like the Agaricea species. But on the next slide, you will see a visual representation of just a fraction of what those species look like once they've been affected by SCTLD. So next slide, please. And it's a difficult thing to experience on a regular, but there always needs to be hope. So with that in mind, on the next slide, we went forward and with our expansion treatment, we were able to work with ocean alchemists to start testing amoxicillin and base 2B in the field. So we treated 250 colonies across, it was actually 7 different sites, that was a typo on my part, in 3 geographical locations. Our highest treatment success rate was with the Orbicellas, specifically Orbicella annularis, and I think as most people, the dendrogyres or pillar corals seem to be the hardest ones to treat. They tend to release a lot of mucus and their constantly extended polyps sometimes make them difficult to have the ointment adhere. But over 8 months, we had really high, up to 90% success rates. And this is what prompted, on the next slide, please, for the government to give us an expansion treatment. So since we received that, before I go there, this is just a montage just to show you the effect of the treatment that we had here. And this one on the left, I particularly love with this very specific little patch of dendrogyra that was able to be saved through the application of amoxicillin in base 2B. Next slide, please. So we started our expansion treatment, and since we were given this permit, we have been able to treat over 200 large priority reproductive colonies. And we are putting a lot of effort into increasing citizen science and opportunistic monitoring through the use of tags, such as the one you can see here. And this means that we're not going out and rigorously monitoring them as we were with the treatment trials. Next slide, please. Of course, antibiotics are nobody's first choice, but as we all know, this disease is incredibly time sensitive. And we have seen a huge reduction in hard coral cover. A paper published by the School for Field Studies in South Caicos, and although there was less dense coral cover in this area pre-SCTLD, they still recorded 62% hard coral cover loss attributed to SCTLD in 2 years. It's not unrepresentative of what I've seen on the other 4 reefs of the Turks and Caicos Islands. Unfortunately, however, without the hard data, this is just anecdotal. But the search for non-antibiotic treatments remains. So we have been testing the new Coral Cure D by Ocean Alchemists. It's a proprietary formula, definitely has a very heavy cinnamon component, smells a little like Christmas, and has so far been giving us some interesting results on a limited test trial due to funding. So on the next slide, please. All these results are based on a single application, whereas all our other test trials had multiple applications of treatment for reinfections. Perhaps our results may have varied, but I think that single treatment results are very compelling for widespread treatment of priority colonies and allowing for just going out there with the hopes that, well, we've got 80% that's going to work. So there are 2 application methods, both in a paste.
1 minute warning, Alize. Sorry, 1 minute.
All right, moving on very quickly to the next slide, please. We had some pretty great success rates, which I'm happy to talk about with people afterwards, but I would like to move on to the next slide to quickly let you guys know a little bit about our outreach and education. So I'm going to go several slides forward, I'm afraid. One more slide. And another slide. And so since 2019, we've done quite a lot of, that's perfect, thank you. We've done quite a lot of different things. And one of our most successful ventures has been working with liveaboards to create meaningful tourism itineraries. And on the last slide, on the next slide, please, you'll see that our first rum-powered research expedition to East Caicos, one of the largest uninhabited islands in the region and anthropogenically unaffected reefs, just came back about 3 to 4 weeks ago. And it gave us an opportunity to collect data on whether these reefs are seeing a difference in the susceptibility of corals and the spread of the disease. So we are still working through that data, but hopefully for a different presentation, we'll be able to give you some information on how those reefs are faring. And on that note, thank you so much, and happy to take any questions.
Thanks, Alize. And just a reminder, everyone can see Alize's full presentation on Whova. And I'm sure we will have some questions for you after we hear about the other response efforts in the region. So our next presenter is Carlissa Callwood from the Perry Institute for Marine Science in the Bahamas. Carlissa.
Thank you. Thank you. Hi, everyone. I'm Carlissa with the Perry Institute for Marine Science. We can move to the next slide. The work I'll be speaking on today came out of the desire for the National Stony Coral Tissue Loss Disease Task Force to create communication tools focused on slowing and stopping the spread, as well as gaining support for crowdsourcing reports of disease sightings. For this particular project, we focused on recreational divers. I'll also talk about how we use the value belief norm theory as a framework to understand what drives motivations. Next. The VBN theory highlights how pro-environmental behaviors, including conservation actions, are more likely to occur when a causal series of variables, specifically values, beliefs, and norms in that order, is present. Values are the criteria that people use to justify actions and to evaluate other people and events. Behavior is thus triggered when an individual comes to believe that their values are threatened and that threat can be relieved by a certain actions. Beliefs are tied to a person's worldviews, whether they are aware of the consequences of a certain threat and how they ascribe responsibility to dealing with it. And personal norms refer to that internalized sense of obligation to act in a certain way. Norms are activated when an individual believes that violating them would have adverse effects on their lives or the things they value. It's also been shown that personal norms are influenced by social norms. That is, individuals are more likely to engage in certain behaviors when they believe the people in their circles value those actions too. Next slide. To use the VBN theory, we created a model that was specific to the disease and added related statements to the survey that was shared with divers. A chi-square test for independence was used to do some very simple stats looking at the relationship between the VBNs and the intended actions. So next slide, what were our key findings from these 64 respondents? Next slide. First, respondents all had a general understanding of the disease and recognize that it's killing corals quickly. Most of what they've learned has been via social media. Next, less than 20% have had the opportunity to help identify and report the disease. And next slide, most are also disinfecting their gear after each trip. And this was true for all of those folks who also worked for dive shops or owned dive shops. Of the remaining respondents, the majority are planning to do both of these actions. Third, next slide, the data shows that no more than 30% of respondents are currently doing either of the boat-specific actions. So those who aren't participating in these activities, such as discharging or releasing their ballast or disinfecting their bilge, are also less likely to commit to doing them in the future. Next, key findings 4 and 5 are both related to motivations. Most respondents have strong values, beliefs, and personal norms surrounding corals and the disease, and these will also serve as strong motivators for them to engage in actions related to halting the spread, particularly disinfecting their gear or reporting sightings. Let's take a closer look at this. Next. When a respondent agrees with a statement, it indicates they have a strong inclination towards that construct. We can see from the results that these divers have strong values related to the disease as a threat, strong beliefs related to personal responsibility and their ability to have an impact, and strong personal norms related to having an obligation to helping. On the other hand, social norms were not as high. Next. Taking a look at the chi-squared analysis, we do see that there is some statistical significance between the strength of VBNs and doing or committing to doing the diving specific actions. For instance, respondents who strongly disagree that they can make an impact are statistically more likely to plan not to disinfect their gear.
It was also interesting to note that those who gave neutral responses were most likely not to participate or commit to taking action in the future. There may be an opportunity here to target these folks who sit on that neutral bubble. Next. However, as we saw on the last slide, respondents were less likely to agree with norms that speak to social pressures. And these norms also did not result in significant differences among responses related to actions. But we should consider that what we see in these responses related to social norms may actually reflect low knowledge about the disease within the general public. And our last finding, next slide, dive shops already include threats to corals in their oral briefings, as well as encourage divers to look but don't touch. However, they don't tend to focus much on conservation and impacts. Next slide. So when we think about communications for this issue, we should be focused on the following. Next. Content related to the disease should be promoted on various social media outlets. This is how most people in this target audience are receiving their information. Also consider sharing content more widely, like through news or radio, to tap more general audiences. Diver awareness is increasing because they spend a lot of time in the water noticing the changes. The same can't be said for boaters or others who spend most of their time on land and don't necessarily see the impacts. Communicating more broadly will assist with this and will also help get at those low commitments to social norms. Content should also be focused on describing the impacts of the disease. As we know from VBN theory, people need to not only be aware of the threats, but the consequences of allowing it to persist in order to be motivated towards action. Next slide. Next, it's important to create general messaging that reinforces people's ability to have positive environmental impacts and personal obligations to help. Content should also further emphasize that look but don't touch mentality, but couple it with info on the high mortality rate and the fast spread of the disease to convey that there needs to be a high level of concern from all of us. And lastly, there should be an increased focus on messaging to boaters and how their actions are just as important. Next. Here are some of the implications. Thank you. Here are some of the implications to be aware of for the wider response. So these particular findings are very specific for divers in the Bahamas, and it's important to remember that cultural context matters. And we also need to acknowledge that we create filters through our own cultural lenses as well. That being said, this does provide the opportunity to conduct similar studies with additional Bahamian stakeholders, as well as audiences in other locations impacted by the disease to provide a broader scope for motivations. And with this information, we can create targeted audience-specific communications to further inspire action. Next slide. We also need to reinforce the impacts of the disease, but that there are actions that each of us can take to contribute. But we also need to keep in mind the number of messages on each communication product and where those products are featured or posted. For instance, the last dive shop I visited had the poster next to the cash register, as opposed to near the tanks where rinsing and disinfection took place. So we have to question, was this poster really accomplishing the intended outcomes? And this speaks to the last implication. Evaluation really needs to be an important aspect of this work. So we need to understand what the outcomes are, how we're going to measure that, and what the strategy will be for improving and iterating on these products based on that. Thank you.
Excellent. Thank you, Carlisa. Moving on next to the U.S. Virgin Islands and Sonora Mealing at the University of the Virgin Islands. Sonora.
Morning, everyone. I'm the marine disease specialist at the University of the Virgin Islands, and today I'm going to be talking about our most recent monitoring program that we established, specifically to look at the small-scale epidemiology of stony coral tissue loss disease. Oh, next, sorry. This disease was first observed on the south side of St. Thomas in January of 2019 at Flat Key Reef, which is an offshore reef denoted by the red star. Next. It quickly progressed throughout the territory, which you can see depicted by this expanding polygon. All of these surveys were conducted by volunteers, both from the university and from the public, so that we could get this very descriptive expansion throughout the territory. The dark pink area shows the total area of extent from previous surveys, and the light pink area shows the area of progression. Exactly 1 year after the initial emergence, it reached the easternmost island of St. John, and just about a year and a half later, it reached the southernmost island of the territory of St. Croix and began to progress throughout the British Virgin Islands to the east. Next. Until today, where no reef in the territory has gone untouched by this disease. Next. Shortly after the emergence of this disease, we established the monitoring program that I referred to earlier. Next. 6 of our sites are located around St. Thomas. Next. 2 are located around St. John. Of these 8 sites, next 4 are actively treated by our strike team divers with the use of amoxicillin and base 2B. At all of these sites, there are between 3 and 7 radial transects, and all of the sites were established before the disease made it to the reef so that we could compare the impact on the community composition before and after the emergence of SCTLD. Next. Next. At the center of each of these radial transects, there's a marked colony of Copophilia natans. Each of these radial transects spans out to 3 to 5 meters in maximum radius. It depends on the site and the coral cover and what we're feasibly able to do in a dive. Surveys on these radial transects include colony counts in binned meters radiating out from that center colony. These are conducted every 6 months after the initial emergence of the disease. Other surveys include health surveys on just the diseased or apparently diseased colonies following a modified agro protocol. Those are conducted at regular intervals as well, but much shorter timeframes after the initial emergence of the disease, including 1 week, 1 month, 6 weeks, and then 6-month intervals following that emergence. In addition to coral-specific surveys, we also conduct video transects along the full diameter of each of the radial transects so that we can extrapolate the benthic cover and community composition. Next. And so far what we found looking just at prevalence, there's a significant difference among sites. Next. The sites that are regularly treated by our strike team divers are denoted by the blue lightning bolt. Next. The site with the highest overall disease prevalence is Buck Island, which is an offshore reef on the south side of St. Thomas. This site specifically is on the sloping portion of the reef and is at a mid-depth range. It used to have really high coral cover and was one of my favorite sites to dive before the disease. Next, the site with the lowest overall prevalence was Megan's Bay, which is on the north side of the island. It's in a very deep bay, and the monitoring site is specifically located on a shallow fringing reef. On average, peak prevalence occurs at time point 3, which is 6 weeks after the initial observation of disease, with the exception of these 2 sites in the blue boxes, Cocculus Rock and Megan's Bay, where peak prevalence doesn't occur until time point 4, which is 6 months after initial observation of disease. It's interesting that the disease took this much longer to peak at these sites because both of them are nearshore fringing reefs with poor water quality and are notorious for being pretty turbid. Next. In addition to site-specific differences in disease prevalence, there's also species-specific differences in disease prevalence. And those differences nicely group the different species into the susceptibility categories that at this point are defined by multiple regions. Next. Those species with the highest overall prevalence include the highly susceptible species. Next. The moderately susceptible species. Next. Phragis astroides, the low susceptible species. Next. As well as 2 species that are pretty rare, so they're not defined by all regions within this stony coral tissue loss disease susceptibility hierarchy. Starting in the top left, we have the agaricid species, which based on their overall prevalence, and from this data, we would suggest that they should be categorized as moderately susceptible. And then down to the right, the Mycetophilia species have extremely high prevalence throughout the first month after the infection or the emergence at the site, suggesting that they should be categorized as highly susceptible. I'd like to include the caveat that this only includes a couple colonies of Mycetophilia, again, because they're a rare species. Next. But the interesting question to most people in this session is, how does disease prevalence compare between the treated and the non-treated sites? So the left portion of this figure shows the non-treated sites, disease prevalence over time, and the right portion of this figure shows the treated sites. Unfortunately, there's no difference in the disease prevalence over time between these 2 different categories. This is true whenever you analyze the prevalence on all species on the reef, as well as only the species that are specifically treated by the strike team divers. Next. However, all the time, resources, and energy spent by our strike team divers is not in vain, because whenever you look at the Orbicella species, there actually is a difference in the disease prevalence over time. I'd like to add the caveat that there's not a difference in the overall disease prevalence, but next. Whenever you look specifically at time point 2, there's significantly less disease Orbicellas on the treated reefs compared to the non-treated reefs, suggesting that the greatest impact of these strike team divers can be seen within the first month of emergence of this disease at a site. Next. However, as you could anticipate from the methodology of this monitoring program, we can extrapolate a lot more data to evaluate the impact of this disease beyond just disease prevalence. So further analyses that I didn't have time to include in this presentation include comparing the coral community before and after the emergence of the disease, as well as through time, as well as the benthic composition. We know that all these corals are dying. There's this new reefscape that's becoming available, so we want to analyze what's actually recruiting to this newly available space. Going back to analyzing the coral community impact from this disease, we want to look at how this might be affected by different lenses, such as the proportion of susceptible species on a reef, the initial density of corals, the average colony size, again, comparing the treated versus the non-treated species, and then the potential role of temperature. So thank you. I look forward to your questions.
Thanks, Sonora. Okay, everyone, start providing those questions in the chat, and Emma's going to start tracking them and lining them up for the discussion that will happen after our next 3 speakers. So now headed to Florida, the origins of stony coral tissue loss disease, and we're going to hear from Nate Burkepile of Florida Fish and Wildlife Research Institute. Nate?
Great, thanks, Dana. Good morning, everyone. My name is Nate Burkepile. I'm a coral biologist with Florida Fish and Wildlife. Today, I'm going to give an update on our Florida Coral Rescue Project and some of the research we've been doing. Next slide. For a quick recap of SCTLD, we know that it's an infectious waterborne disease with high rates of disease transmission and mortality. SCTLD has also significantly impacted at least 22 Florida coral species. Next slide, please. In the fall of 2014, SCTLD was first observed near Virginia Key in Miami-Dade County. From then on, it has rapidly spread northward and southward. Unfortunately, today, SCTLD is currently making its way through the Dry Tortugas National Park at the end of the Florida Reef Tract. Next slide, please. The Coral Rescue Project can be defined as a multi-agency, multidisciplinary effort to collect SCTLD susceptible species from the Florida Reef Tract to hold and preserve genetic diversity for future propagation, restoration, and reintroduction to the wild. Next slide, please. Our collection plan was put together in 2018. Prioritization of collection species was based on susceptibility to the disease, the speed of disease progression across the colony, and the prevalence of whole colony mortality. This resulted in 16 high priority species and 4 medium priority species. We also developed a genetic management plan to guide these rescue collections and ensure that sufficient genetic diversity would be collected to support restoration activities. The genetic target number for restoration is 50 unique individuals, or genets, per rescue species. This target would need a minimum of 200 individuals per species to be collected as geographically spread out as possible. This plan includes the development of genetic markers for all 20 rescue species, so all corals are genotyped. Lastly, this plan includes a connectivity study for brooder coral species and the development of a genetic catalog to house all of the genetic information collected during rescue activities and make it available to the broader restoration community. Next slide, please. FWC hosts a public online dashboard for coral rescue data viewing and tracking. The most recent update for this includes 20 species under care at 26 facilities across 14 states. FWC is also currently in development stages of a new data portal, which will help track individual colonies. Next slide, please. With that, it's also important to note that the many organizations that make this project happen. The Association of Zoos and Aquariums, AZA, has contributed millions of dollars of their own. Those facilities house many of the rescue species. Next slide, please. Dendrogyra has now also been incorporated into the rescue project. There are approximately 535 colonies between aquaria. With the unfortunate movement of SCTLD into the dry tortugas, and specifically observation of tissue loss due to SCTLD on wild colonies, 4 dendro fragments were collected and transported to Frost Museum of Science in Miami. Frost currently has 116 dendro fragments. Next slide, please. Rescue corals have also now been in aquaria long enough that we can learn from their propagation and use the spawn for restoration projects. A few interesting points to note include 11 species at 7 different facilities have now spawned. Florida Aquarium has been a huge part of this propagation success, where they have sent over 500,000 larvae to 7 institutions. Lab-induced spawning, where all variables are in a controlled setting, has occurred for 4 species. 9 rescue species have been raised from sexual recruits to 1 year plus post-settlement. And finally, wild gametes from Diploria labyrinthiformes have been cross-fertilized with ex situ gametes of rescue corals. The larvae of this cross-fertilization has been settled, and now the recruits are being reared. Next slide, please. Yes, the genetic management plan was developed to guide coral rescue collections and ensure that sufficient genetic diversity would be collected to support restoration activities. Currently, we have completed our first discovery panel. This includes MIA, DSTO, EFAS, PSTER, and DLAB. The markers were made by GBS, which is genotyping by sequencing, if anybody was interested. Next slide, please. So for our first discovery panel, we have found high genetic variability in all samples, and we reached our goal of 50 unique genotypes for this panel. EFAS is the only one to have clones. In total, 4 of the genotypes out of 230 had clones, one of which is represented by 4 different colonies, and the other 3 are represented by 2 different colonies each. Interestingly, these colonies are also found within 2 miles of each other in the dry tortugas. The genetic threshold for calling clones is 99%, whereas the other species, DSTO, DLAB, PSTER, and MIA, can show up to 98% similar genetics and still not be considered a clone. DSTO samples showed no clones, but some very closely related genotypes. So the number of SNP markers are being doubled for a more detailed analysis. Next slide, please. We're currently in process working on the second discovery panel, which includes MALI, MLAM, OAN, PCLE, CNAT, and Dendro. Once we have completed this panel, then all genetic markers will be made publicly available, which is useful to compare relatedness between genetic samples. Next slide, please. So now we get into some of our research projects. The first project I'd like to go over is our brooder connectivity study. This study was put together so that we can understand the connectivity of some of these brooding species, which ultimately helps us understand population management for good restoration practices. So we took genetic samples to assess connectivity of 3 breeding species, that's EFAS, MALI, and Porites astroides. Porites is not a rescue species, but is included in this study to fill some spatial resolution gaps since we have previous genetic information about this species. We have collected 345 samples to date between the dry tortugas, Marquesas, the Lower Keys, and Southeast Floridas. However, those samples have not yet been processed. Next slide, please.
2-minute warning, Nate.
Great, thank you. A trio outplanting study was conducted to assess whether stakes excluded predators from the outplanted corals. We outplanted 440 corals at an inshore patch reef in the middle Florida Keys. The study design included 3 different species, CNAT, DLAB, and MLAM, of 2 size classes, small and large. These 3 species were babies settled from coral rescue species parents at Florida Aquarium. There were 2 predator treatments and 1 control, which included no stakes, were dispersed at various colonies. The predator treatments included crowns, outward-facing stakes, and tents, inward-facing stakes. Monitoring time frame included a 2-week check and then a monthly status check from then on until 1 year. We found 76% total survival of all coral species outplanted. That's 334 of the 440 are still alive to this date. We found 95% survival within the tent treatments, 79% survival within the crown treatments, and 71% survival within the controls. Next slide, please. We also found that predation declines over time. So as we approached our 1-month outplant monitoring, we noticed no new mortality from predation. Predation mainly occurred from fish, where you can see the scrapes here on the tile. This study helped put together a new outplanting study that we refer to as the acclimation study. This will include only CNAT and DLAB babies from Florida Aquarium, and will look to see if predation pressure is relieved after 1 month. To do so, we have corals in situ and ex situ nurseries where they will sit for 1 month and then be outplanted. Next slide, please. Really quickly, by May of 2020, 7 liveaboard cruises were conducted and collections ahead of the disease boundary were completed. Furthermore, by the beginning of August 2020, we moved back to conducting single-day collections within the disease endemic zone, followed by land-based processing. These endemic zone collections focus on rescuing survival corals in advance of coastal construction projects. The corals surviving in the endemic zone represent potentially resilient genetics and can be used to incorporate into future propagation activities. Next slide, please. And finally, since we did not reach our 200 colonies collected, we will be conducting supplemental endemic collections, which includes the species listed here. And then last slide. Yep, and thank you for taking your time to learn about the Florida Coral Rescue Project. If you have any questions, I can discuss in a little bit, and you can send them to my email here.
Thank you, Nate. Moving on to Kelly Pitts from the Smithsonian Marine Station. Kelly, talk to us about probiotics.
All right. Hi, everyone. I'm here to talk about our use of probiotics in the Florida Reef Tract. Next slide. So before we begin, I just wanted to make sure everyone's on the same page. So I thought I would define probiotics. And according to the World Health Organization, probiotics are live microorganisms, which when administered in adequate amounts, confer a health benefit on the host. So I think we're all used to probiotics that you use for humans, like daily supplements, but probiotics have also found a large space in the world of aquaculture. And because of that, Dr. Blake Ushijima thought maybe we could use probiotics to treat corals with stony coral tissue loss disease. Next slide. So far, that started our search for our probiotics. And so far we have 2,000 isolates that we've isolated from healthy or more disease-resistant species. You can see all 7 species listed there, all from the Florida Reef Tract. And we found 400 of these 2,000 isolates that display antibacterial activity against putative pathogens. So you can see on the right, we have putative pathogens plated out on the Petri dish. And each of those isolates are streaked out. And if they show this zone of inhibition or this clear region around them where they're inhibiting the growth of the putative pathogen, we consider them that they might be a good probiotic. Next slide. So far we found this one promising strain, MCH17, which is a Pseudo-Altermonas species that's isolated from Montastria cavernosa. You can see it on the left in yellow. That yellow spot there is MCH17, and it has a very large zone of inhibition where it's stopping the growth of the putative pathogen around it. And it does this by creating these 3 different antibacterial compounds that you can see on the right. So it's a pretty powerful strain. Next slide. So now that we have MCH17 in mind, we needed to test it out in the laboratory before we bring it out into the field, just to make sure it actually can stop the disease. So we took 22 species of Montastria cavernosa and broke them into 2 different pieces. We treated one half with filtered seawater and the other half with MCH17. And you can see that disease arrested on 55% of the colonies treated with MCH17, and disease slowed on an additional 14% of those colonies treated with MCH17 compared to the controls. So it looked pretty promising in the lab. Next slide. So the next step was to bring the probiotics out into the field. And this is the first time this has ever happened. So we weren't really sure how to do this. So we had to get a little creative. Next slide. So we have this plastic bag treatment. It's a whole colony treatment where we put the plastic bag over top of the colony, and it has weighted line along the bottom of it to keep it in place. Next slide. We inject our MCH17 from a syringe through aquarium tubing up underneath of the bag and leave the bag for 2 hours to allow the bacteria to colonize on the coral before we bring the bag back to the boat. Next slide. And this is a pretty cost-effective treatment where we can treat each colony for about 27 cents. Next slide. So we also have a lesion-specific treatment, which is, go ahead, next slide. Sorry, this is all one by one. But we use a paste that is filled with our MCH17. It's kind of like a toothpaste, and it polymerizes or thickens when it's in contact with divalent ions in the seawater. So it actually, it's also non-toxic to the probiotic, which is really helpful. And you can actually flatten it across the colony so that it won't kill the tissue underneath, but you can make sure the probiotic actually grows on the coral. And again, this is pretty cost effective where you can treat each colony or 3 to 5 colonies, depending on the length of the lesion, for about 24 cents. Next slide. So that now that we have our treatments, we had to set up a site to test out these treatments. So with the help of Dr. Brian Walker's team from Nova Southeastern University, we set up this site off the coast of Fort Lauderdale, Florida. It runs north to south, and you can see on the left, all of those numbers represent different Montastraea cavernosa colonies that were treated with one of 5 different treatments. So you can see we have a probiotic bag, a probiotic paste treatment, each of their controls, and then our background controls where we're just monitoring the colony over time. Next slide. So this is our timeline for that site. We took tissue and mucus samples for genomics and metabolomics analysis, and we just published a new paper that you should definitely check out on the metabolomics from these colonies. We took photographs each time we went of each colony, and next slide, you can see we treated the site 3 different times. And if the coral got 1 treatment the first time, we continued with that same treatment each time we were at the site to treat. Next slide. So these are some of our initial observations. This one's pretty exciting. You can see the colony on the left here looked like it was way past the point of any return, and we've treated it with probiotic paste. And you can see in just a few months that some of those polyps that looked like they were about to die actually regained zooxanthellae pigmentation, and we call this healing, which is pretty exciting news. Next slide. This is also another initial observation we saw. We had to kind of work out the kinks in our methodology, but this is a picture of a colony treated. This is 2 weeks after being treated. Next slide. I think
2-minute warning, Kelly.
Thanks. Next slide. So we treated it, and you can see the probiotic was actually put on the left side of the colony because that's what looked the worst. Whereas it's really important to make sure that probiotic makes it across the entire colony to treat the whole coral. Next slide. So we binned these colonies into different classifications based on disease severity. We had healing, which was bleached tissue that was regaining pigment, stopped diseased, slow or quickly progressing tissue, which is chronic or acute tissue loss, or colonies that have died. Next slide. What we found was our probiotic bag had 6 out of the 8 colonies that were healing, which is great news, whereas our probiotic paste had 2 colonies that were healing, but we weren't sure what to do with that because the control paste also had 2 healing colonies. Next slide. So next slide. We have this 3D model that we could create of each colony, and you can, using the software, measure the healthy tissue remaining on each colony over time. Next slide. Thanks. So this is our healthy, or this is the tissue lost over time. You can see the 3 arrows at the bottom represent our treatment days, and our probiotic, or our background controls significantly differed from the other treatments, whereas none of the other treatments significantly differed from one another. We think that's because we have a smaller sample size. So now we have 3 more sites of corals we probably have over 100 colonies that are treated now. So we have to get all of our results from that to add to this to really tease apart some of these differences. Next slide. So this is our healthy tissue lost over time. So the graph before was just total tissue loss. This is only considering healthy tissue lost over time. Again, I have those 3 arrows to represent our treatment days. And from this, you can see that it appears that the probiotic paste and probiotic bag treated corals were losing less healthy tissue over time. So from the previous graph, you can see that those probiotic treated corals were losing tissue, but from this, it goes to show that it was tissue that was already diseased. Next slide.
Kelly,
So
we're
with
gonna
that,
have to Oh, perfect.
Yeah, sorry, just wanted to thank everyone. You can see how many people are on this project. And next slide, how many collaborators we have. And with that, I'd like to accept questions in a few minutes.
Perfect. Thank you.
Thanks.
Hold your questions for Kelly because we have one more presentation to hear about, and then we will entertain all ideas and questions and comments for our second panel. So to wrap up the session on stony coral tissue loss disease, we now have Sandra Mendoza Quiroz from Seacor International. Sandra.
Hello, everyone. Can you hear me? Thank you.
Yeah.
I will talk about a case study on the offspring of Pseudodipleuris strigosa affected by stony coral tissue loss disease. Next slide, please. The first records in the Mexican Caribbean of the outbreak of this stony coral tissue loss disease occurred in the summer of 2018, which affected 22 species of the corals and in 82 reefs. And in the first study of this outbreak in Quintana Roo, notes that the Pseudodipleuris strigosa was the second species affected, most affected in the region. Therefore, next, please, the prevalence of this species on the reef and its reproductive success is a concern. Next, please. Thank you. Other, please. To follow, sorry. For this reason, our objective was to follow the survival of the offspring obtained from a colony affected by stony coral tissue loss disease, crossed with a healthy colony, 100% apparently healthy tissue of this species. And next, please. To achieve this, we collected gametes from these colonies during the spawning event in August of 2020 at Buchanan Reef in Puerto Morelos, Quintana Roo. The collected gametes were taken to the lab for cross-fertilization for 1 hour. Later, the eggs were rinsed and placed in static cultures using seawater filter to 1 micron and UV sterilized. When the larvae formed, the artificial substrate with, designed by Seacor International and previously conditioned in the sea were offered to the larvae for settlement, and the settled larvae were counted 14 days later. The settlers acquired symbionts naturally from the water and substrate provided without direct inoculation of a specific symbiont. We obtained the settlement yield that was defined by Miller and collaborators as a proportion of the initially obtained embryos converted to the all-plantable settlers. Next, please. Okay, what we've when the settlers, we've also 1 month. Sorry, we transferred the settlers to the aquarium to be cultured under controlled conditions of light, salinity, and temperature. We feed with different supplements, and we monitoring the survival of these recruits at 4 and 12 months of age. Okay, we transplant these recruits to the reef in a degraded reef of Puerto Morelos with presence of the healthy and diseased colonies. And we monitoring the survival of their 15 and 30 days after the outplant. Okay, what we found is that the gamete fertilization was 95% after 2 hours, and it was verified by observing the first cell division of the embryos. And the settlement yield obtained was 22.39%, higher than next one, please, higher than the obtained in another cross of disease colonies with ACTLD and black band, and higher compared to the settlement yield of recruits obtained from a healthy colonies of the same species in previous year. And next, please, higher compared to other studies from other regions of the Caribbean and that use the same time of substrates. Next, please. Post-settlement survival showed a drop from 100% to 7.8%, and it should be noted that the intensive care was at time discontinued due to the impact of 3 hurricanes in Quintana Roo coast during the year of the rearing in aquarium, specifically at month 2, 3, and 12 of age of this course. Next, please. And the growth in this period was from 2 polyps to 6 polyps per colony. The survival planted on the reef decreased from 100% to 38% after 1 month, mainly due to the predation of the fish by competition with turf algae. And from these findings, sorry, next, please. Thank you. For these findings, we conclude that the cross between a healthy and diseased colonies produce a healthy offspring with very high fertilization and very good settlement, which indicates that there is apparently no transmission of the disease. We found also the settlement yield was high when compared to other healthy colonies obtained in different years of different Caribbean regions. Finally, it shows that the assisted sexual reproduction process can potentially be used in rescue of some species affected by stony coral tissue loss disease, even if the colonies are diseased. Finally, next one, please. I would like to thank the people and institutions that have made this study possible, and I will be happy to answer your questions. Also, in the next slide, there are my contact information in case you want to send me some email. Thank you.
Perfect, right on time. Thank you, Sandra. Okay, the moment you've all been waiting for. Time for questions, discussion, share your thoughts, reactions. Emma, handing over to you to facilitate that.
Okay, so we had, let's see, we've got some questions for Carlissa and for Alize. Let's see, let's go to Carlissa. Are you there? We've got a question that came up in the Q&A. Could you have answered that already? Let's talk about that. There was a question saying that the Florida Keys have a Blue Star program that lets customers know if dive operators are committed to coral conservation by educating divers and snorkelers about better diving etiquette. Could a similar certification program focused on education and sanitizing dive gear be helpful in preventing the spread of stony coral tissue loss disease? Would you like to give us any guidance on that, Carlissa, based on your research?
Sure. Yeah, I definitely think a certification program like that would be helpful and useful. It would definitely help to get most of the dive shops on board with talking about the disease and talking about the impacts, especially while they're on the boats, which is when we really need them talking about it and encouraging the disinfection. And I feel like certification programs like that also help with being revenue drivers as well, because divers tend to be really keyed into those eco-friendly operations, and those are the things they want to give their money towards.
Okay, thank you. There was also an interesting question in the WOVA chat asking you about, let's see, this was from Emily Melvin, who was asking you about the culture of disinfecting gear and to what extent that links with the beginning of the pandemic. And what helps prompt the divers into this behavior?
Yeah, I saw that question on Huva. I definitely think that the culture of rinsing gear and disinfecting gear is something that is already part of the dive culture. Yes, there was some of that, an increase seen with the pandemic, but it was something that was already happening prior to that. But I think most importantly is that for those people who don't currently disinfect their gear, knowing that it's something that happens on a broader scale and knowing other people that do it will be motivations for them to add that to their behavior moving forward.
Okay, perfect. Thank you. Now, Alize, you showed interest also in a standardized certification program, so that might be interesting to keep in touch with Carlissa, with Ellery. But let's come back to you, Alize, because you were presenting some results about the Christmas cure, the coral cure, CCD, and about the effectiveness of the hemp rope. Would you like to just summarize those results for us? And if you want Caroline to pull up a slide, maybe she can do that.
Or I can, does that work?
Yes, it says your screen sharing, double click to enter full screen. Let me see. Okay.
Okay.
Okay,
So,
in just less than 5 minutes.
yeah, so basically the rope and the paste had similar success rates on that one transect with only one application, 68% success rate versus the 80% with amoxicillin, but it raises some interesting questions about then the desire for non-antibiotic cures as well as regulations in some countries that would allow for that to be more easily used. We had another, so here you can see we did, and this was a small colony, but we also did treatments on Orbicella annularis with genetic comparisons that we're still going through the results of with the base 2B and the Coral Cure D paste. And the paste was more effective on some of the other transects, but overall it was promising and definitely makes us want to do a few more test trials to bolster those numbers. Yeah.
Okay. All right, thanks very much. Now, last chance. Are there any other questions for any of our panelists, presenters, any of the other experts in the audience? We've got a really great crowd here today. Thank you everyone for joining in. Any last questions? If not, going, gone. Over to you, Dana. Oh, yep. Here we go. There you go.
Thank you so much, everyone, for just sharing all of your great research and effort. It's been a great community of sharing our findings and our lessons learned, our mistakes and successes with stony coral tissue loss disease. And just thanks so much for all of your work and for sharing that today to our presenters. I want to encourage everyone to also go visit the posters. We have 8 different posters touching on stony coral tissue loss disease efforts. You can visit those in the HUVA application, ranging from research efforts to response efforts all across the region. There's so much good work going on. As Emma mentioned in the beginning, we do have a bi-monthly meeting that is open to partners working across the region on stony coral tissue loss disease. Judy Lang and I facilitate that group. And if you are not already engaged and would like to be, I posted our emails so that you can let us know and we can pull you in. Emma, it does look like we did get a question in from Alfonso Aguilar, who has a question for the panelists, and I'll just go ahead and read it. He wants to know if column sediment or diver gear are possible vectors of stony coral tissue loss disease. And how is it possible that the mechanical application of amoxicillin to coral colonies may prevent the spread of SCTLD? So I don't know if anyone wants to take that question. I feel like we have had some related questions earlier.
Michael has his hand up.
Go ahead,
I'm happy
Michael.
to take a stab at it. So I wonder if it might be a question of a pathogen load here, where a lot of the suspected vectors or reservoirs have relatively lower prevalence in it, such as like in sediments. The particular pathogens might not be all that abundant, and it might take some type of concentration. And so in a system like that, treating infected corals that have a much higher pathogen load might actually help to reduce spread. It could also be a question of where these communities are found, whether they're found in, say, the boundary layer of sediments or on the boundary layer in the water between the coral colonies. It might require some type of perturbation to make those microbial communities then exposed to the coral. So it's perhaps not just this constant bombardment of pathogens, but might be a key set of factors that are influenced by the surrounding environment that determine whether there is disease spread among colonies. But arguably, this question has not been well addressed because it's very difficult to answer it. It requires examination of multiple spatial scales, and perhaps some of the modeling approaches that have been implemented can start to look at some of these microenvironmental variables that might influence how an individual colony is transmitting disease to another. I think Sonora has her hand up as well.
Yes, Sonora. Thank you, Michael.
Yeah, just to add on to Michael's, I guess, explanation that there's probably a pathogenic load. We've run some transmission experiments that suggest that that is true. It's not a binary exposure or not to the pathogen. There actually is a threshold where the corals can resist gross lesions. And then on his second point about how we need to tease apart with modeling, Louisiana State is working on that with the data that I presented from the radial transect. So they'll be doing, I think it's SSR models. So they'll do within reef distribution of the disease, but then also geographic around the entire territory. So stay tuned.
Thank you, Sonora. Any final questions or comments? Any thoughts from my co-hosts here, Emma and Caroline?
No, thanks, Dana. I think we're right on time. Great session, and this was really, really well attended. Thanks for all the great abstracts and all the great presentations.
Great. Thanks, everyone. We can give our translators a little break before they have to jump to their next session. And thank you so much.
Thank you, everyone.
Yes, thank you to all our panelists today. And as Dana mentioned, we're going to have a short break now, and we will reconvene at 12:45 for our Sargassum session. Thank you, everyone.
Thanks, everyone. Great session.
Hi, good morning, good afternoon, good evening, everyone. Thanks for joining us and welcome to the Sargassum session. We're pleased to have you, and we have such an impressive lineup from well-established researchers to early career ocean professionals. And you may have seen some of the presentations before, but this afternoon, each panelist would be able to present 3 to 5 minutes around their presentations. My name is Shelley Ancox. I forgot to introduce myself there. And of course, with me joining my esteemed moderator, Hazel Oxenford. It gives us great pleasure to be able to moderate this session. We've been working on Sargassum for a decade, actually. I remember when it first came on stream, I was in some SEAG communities in Barbados and St. Lucia, working with divers. And, it was something we hadn't seen before. And it was quite interesting to work with them to see how they cope and adapt to something like this. We are ready and willing. We're just going to do a quick shift in terms of presenters while we get Dawn Johnson to join. So first up is Christy Aline. She's presenting, and Christy is a really good colleague and friend of mine. We spent time working on other things, but I'm very happy to see now that she has advanced in her career and she's currently pursuing a PhD at the Royal Maritime University. And this afternoon, she's going to present for us the title, A Preliminary Assessment of Sargassum Associated Biodiversity, Implications for Management. Christy, the floor is all yours.
Hi, everyone. Thank you, Shelley. First, let me say it's a great pleasure to be part of this year's GCFI discussions. As Shelley said, my name is Christy, and I'm a PhD student at the Royal Maritime University. The submitted presentation was actually a joint effort between myself and Ms. Makeda Corbin, who's an MPhil student at the University of the West Indies Cayfield campus. Our talk provided a preliminary look at the biodiversity associated with sargassum influx events in Barbados. And the work is part of ongoing efforts to identify what biodiversity is associated with pelagic sargassum, and does the biodiversity associated with sargassum coming into Barbados change over space and time. To account for the organisms both within the mat and below the mat, we used a combined dip net and video sampling method at various distances from shore. During our assessments, collected organisms were categorized, as you see in the diagram, as either epiphytic, clinging, or free-swimming fauna. Epiphytic fauna was described as any sessile organism which grows on the sargassum and may include things like hydroids and tube worms and encrusting bryozoans, whereas clinging fauna described organisms that exhibited very limited range of movement and spent their entire time clinging to or climbing around the sargassum, and this may include crustaceans and polychaetes and mollusks. Free-swimming fauna refer to species that swim beneath the sargassum, and this includes fish species and juvenile turtles and adult turtles. Collection efforts for July and August indicated that while a variety of epiphytic and clinging fauna were actually abundant within 0 to 500 meters of the shoreline, free-swimming fauna have appeared to abandon the mat at this close to shore. At the moment, further investigations are required to identify the significant trends in spatial and temporal distribution of the sargassum-associated species, and to better understand what the trade-offs are among social and ecological objectives for beach and in-water removal of sargassum. It is our hope to continue this assessment into 2022, looking at winter and summer influx events to get an idea of what's really happening over a longer time period. Thank you very much for your time, and I'm here to answer any questions.
Okay, that was short and sweet. Thanks, Christy. We're going to hold the questions for the end, but feel free to drop your questions in the chat box or the question and answer box. We'll be monitoring both of them, and we'll also try to monitor the Hoover session as well. Okay, I'll turn over to Hazel to introduce herself a bit more and guide us in the rest of the session.
Thank you very much, Shelley, and welcome everybody. It's a real pleasure to be co-chairing this session with colleagues, friends, and students. And it's quite a small community that have been working on sargassum from the very beginning. And it's a very nice opportunity to hear the progress that's been going on. So I'm going to introduce our next speaker, Karima Degia. She's actually a registered professional engineer specializing in coastal engineering with a focus on sustainable environmental management. She's also a freelance consultant and a research associate with us at CERMES here, the University of the West Indies in Barbados. And she's been an integral part of our sargassum work at CERMES, not only as a sargassum project manager, but also working on many technical aspects, particularly what she's presenting today, understanding and mapping exposure to sargassum and ecosystem impacts. Over to you, Karina.
Good afternoon, everyone, and thank you to Hazel for that introduction. Thank you, Shelley. Thank you for the team in general. I'm happy to be here. Please bear with me a little bit. There may be some background things going on. So good afternoon from Glasgow and in COP26. And thank you to the CARICOM Pavilion for allowing me to carve out a little corner of space to do this here from what is a very hectic venue. But I'm very happy to be here and joining you for this session to talk a little bit about the sargassum exposure mapping that we've been working on. So I think we know that the impacts of sargassum influxes in the region, they've been very well documented, and we don't need to repeat that. We know that there are impacts on tourism, that there are impacts on fisheries, on public health. But those impacts are usually described when we see that in general terms. But there are differences in space and time in those impacts. And we're focusing really here on those spatial differences. And we know that the Caribbean as a region is not as homogeneous as it is often perceived to be. I think that's a really important point. And within that variety that we see in the region, we want to focus a bit here on small island developing states and their unique circumstances within the region that are already well recognized. And the unique challenges faced by Caribbean SIDS underpin as well how sargassum influxes impact us. Sargassum piling up on a small pocket beach on the east coast of Barbados at the very edge of the Atlantic is quite a different matter from sargassum on a very long beach on the Caribbean coast of Mexico. Different in terms of its impacts and different in terms of the logistics of addressing the impacts. And so that's just one example. So what we wanted to do is focusing on the 5 participating countries in the project is to map the exposure to sargassum around each small island. The difficulty with that is that within these SIDS, there isn't an abundance of data, of monitoring data on sargassum exposure or on historic influxes. And oftentimes when you hear about exposure mapping for, say, other hazards, say hurricanes or flooding, that involves probabilistic modeling of flood exposure. That's not an option for us here with sargassum. So we had to turn to other methodologies. And what we did, we went to everyone's favorite space for aerial photography in the public domain, Google Earth, and extracted the time series of photos available for each participating country that showed us when and where sargassum influxes were occurring, and used those aerial images to build up maps of sargassum exposure for each country. We made use of existing coastal zone sub-areas in some countries like Barbados that already have that framework as part of coastal zone management in country, so that we had stretches of coastline that allowed us to generalize within that stretch and to be able to say, well, within this sub-area, exposure is medium or exposure is high. Having done that, we are able to overlay that sargassum exposure with mapping of assets in key asset categories that we're interested in. So for example, we can map tourism assets like hotels and dive sites and dive shops and marinas, and overlay that with the exposure mapping so that we can see spatially where will the impacts on tourism occur within Dominica, for example. We can map fisheries assets, landing sites, etc., and look at where that sector is going to be impacted specifically within each country. We also looked at coastal ecosystems, so we can understand the spatial differentiation of impacts on coral reefs, mangroves, seagrasses. And this is work that we perceive can be built on in a number of ways. Firstly, we would like to see improvement of data collection in terms of sargassum monitoring. That would help to improve the accuracy of the exposure mapping, but we also foresee that this will help with further impact analysis and really differentiating where impacts are occurring, and also planning the response. If there's mapping of where impacts are occurring and what type of impacts, then it will enable planners to know what type of resources need to go where for what intensity and what type of impacts. So that's really what the work has encompassed so far and where we'd like it to go. And I am here as well for any questions or comments that you may have. Thank you.
Thank you, Karima. Very good presentation. We're seeing some questions in the chat box. Feel free again to pose your questions there before we come to the official question and answer session at the end. Next up on our program is Mariana Leon Perez. She is a PhD candidate at the NOAA Center for Coastal and Marine Ecosystems. And she's a scholar at the Hart Research Institute for Gulf of Mexico Studies. So this afternoon, Mariana is going to present under the title, Characterizing Sargassum Accumulations Along the Coastline Using Satellite Data and Google Earth Engine. Thank you. Let's welcome her.
Thank you so much, Shalian. Yeah, so good morning, everyone, and thank you for this opportunity. Today, I'm going to be talking just a summary of the research. This research is part of my dissertation, which focus on especially assessing the socio-ecological vulnerability of coastal communities in Puerto Rico to sargassum influxes. The part that I presented constitutes the exposure component of the vulnerability assessment we are conducting. So very similar to what Karima Deja just said, when we were designing this project, we realized that there was a data gap and that the sargassum detection efforts that were available at that moment focused on offshore waters and not in the coastline where sargassum gets accumulated. So therefore, we decided to address that data gap and develop a method for characterizing sargassum influxes along the coastline. And we decided to use Sentinel-2 satellite data and the Google Earth Engine online platform to process that imagery. So in the middle of the slide, you can see that, that this method was capable of not only detecting fresh sargassum as previous detection efforts have done, but it was able to detect also decomposing sargassum and sargassum brown type. So far, the accuracy of this method is really good, although we are still testing it and we want to also incorporate more field data. The graph below shows an example of a time series from 2015 to 2021 for one of the sargassum accumulation hotspots that we identified in La Palguera, Puerto Rico. So you can see here in the graph that there is an annual seasonality of fresh and decomposing sargassum, which is in blue and white, and that 2018 was the year that experienced the highest area of accumulation. But also very interesting, you can see how sargassum brown type makes a peak few months after fresh and decomposing sargassum maximum. So this tells us that, very similar to observations in other Caribbean coastlines, that in these two accumulation hotspots that we selected, the influence of sargassum, or the exposure, is not only occurring during summer months, but basically throughout the whole year. It could be fresh, decomposing, or brown type that is being present throughout the year. And these have very important implications for the socio-ecological vulnerability of this community. So we are continue working on the code and improving the method so we can hopefully in a near future apply it to other areas. So thank you very much for listening, and please type down any questions you have.
Thank you, Mariana. Great presentation.
Yes, thank you very much. That's really very interesting. Our next speaker is Bethia Thomas. Bethia is currently pursuing her PhD here at Stony, University of the West Indies, on the Cave Hill campus in Barbados. But she's actually a resident in one of the heavily impacted communities in St. Lucia. And she's very passionate about making sure that the voices of community members is actually heard in helping to build resilience to Sargassum influxes. So she's going to talk to us today, or summarize her presentation on the community-based participatory approach to vulnerability analysis and adaptive planning for pelagic Sargassum influxes in St. Lucia, or using St. Lucia as a case study. Her PhD has been quite interesting, having had to do her fieldwork during COVID shutdowns and also the birth of her third child. So good for you, Bethia. Looking forward to hearing you.
Thank you, Professor Oxenford, for your kind words of introduction. Good afternoon, everyone. It is a pleasure to be here to present the preliminary findings of my research at this prestigious conference. My research is in its preliminary stages of data analysis, and so what we will present here to you today will be our initial findings. I am also supervised by Dr. Janice Cumberbatch, who is a senior lecturer at Stony. In the last decade, the shores of the Caribbean have been inundated with unusual amounts of pelagic sargassum. And the first strandings were reported in 2011, and they continued almost yearly, reaching unprecedented amounts in 2018. The record-breaking events demonstrated that greater quantities of pelagic sargassum were affecting the Caribbean region with greater frequency and over longer periods of time. So you have greater frequency, longer periods of time, and longer duration. This research endeavored to evaluate the vulnerability of 3 rural fishing communities in St. Lucia, namely Dennery, Micoud, and Pralin, to extraordinary pelagic sargassum influxes. The study employed a community-based participatory approach and used those approaches to develop 18 indicators for the 3 components of vulnerability, namely exposure, sensitivity, and adaptive capacity. The indicators were then aggregated to form a combined vulnerability index for each community. The research employed a mixed method approach for data collection using both quantitative and qualitative techniques. And what this allows us to do is to synthesize quantitative data with experiential data, which is not numerically quantifiable. So we use the qualitative data to tell the story behind the numbers. And so the methods we used included household surveys and census data to compute the index, followed by focus group meetings with fisherfolk, key informant interviews with policymakers, and the unique feature, community meetings, that I have dubbed the voice of the invisible. And the voice of the invisible specifically targets the poor, disabled, women, and minority groups, which would have not perhaps been previously included in data collection. And as a result, their voices have not been heard and their views have not been captured. The approach chosen for the creation of the composite index is an adaptation of the multidimensional poverty index, or MPI, henceforth referred to as multidimensional vulnerability index, MVI. Now, why poverty index, you may ask? You see, poverty lies at the heart of vulnerability, and while the two are not synonymous, they are strongly linked and often caused by similar factors and processes. Now, MPI is a very useful tool because it measures the proportion of people in a given population who experience multiple deprivations or vulnerabilities in our case. And so we are able to tell who is vulnerable in what indicator and across what components of vulnerability. It has two important features, H or head count, which is the incidence, as well as A or average, which is the intensity. So incidence and intensity. And the MPI is calculated by multiplying or finding the product of H and A. Using our preliminary analysis, we came up with results only for the community of Pralle thus far. And so Pralle has shown an MPI of 53%. Additionally, 21% of the population displayed vulnerability in 8 out of 18 indicators. So most persons were vulnerable in 8 out of 18 indicators. No knowledge of the National Sargassum Management Plan and no income generation from alternative uses of sargassum were the most popular indicators, affecting 97% and 98% of the population respectively. From the community meetings, the residents have developed their problem and solution trees, which they will use as the basis to develop their participatory videos to tell their story in their voice. And fishermen continue to try different strategies to effectively deal with sargassum, but are open to new strategies, including the use of booms, forecasting, and further research. I have a lot more to tell you. However, I look forward to sharing with you the remainder, any additional information you may need during our Q&A or on the app. Thank you for your time.
Thank you very much indeed, Bethia. That was lovely. We're missing one of our presenters and urgently trying to get hold of him. In the event that he's not joining us, I think we should open up the question and answer session. And if he's able to join us, this is Don Johnson, and then we will bring him in as soon as possible. If anybody has any questions for any of the speakers, including Don, if you've watched his very nice presentation, we'll attempt to answer them. I am unable to write anything in the question and answer box. It seems to be locked for me. So please keep the questions coming through the chat. We have our first question for Christy. The question's coming from Mark Peterson. And it says, what % of species in the Sargassum are recreational or of commercial importance?
Hi, Mark. Thank you for your question. Preliminary results is only for 2 months so far, and out of the 26 species that we've identified, roughly 15% were commercially valuable, which would be jacks, and our recreational value would be the sea turtle. But largely, we've only found like smaller stuff like crustaceans and amphipods and hydroids and stuff. The extent to which that serves as a food chain for the commercial valuable, not sure as yet, but to date, only 15% of what we've seen could be classified as commercially or recreational valuable.
Thank you. I thought it was very interesting that you said that turtles were recreational. In a fisheries meeting, that generally means recreational fishery. So please expand on that one.
No, I mean like for the dive industry, just like recreational fishing. So, I mean, sorry, not fishing, not to eat. That's not what I was going for. Yeah.
Yes, I knew really. Turtles have a tremendous value here in Barbados and in the other, in many other Caribbean islands in swim with turtle programs and just in viewing turtles on scuba dives and so on. Thank you, Christy.
Oh, yes, sir. No luck getting Don. I got his voicemail, so I believe we will carry on with the question and answer at this time. There was a question here for Christy. What are the implications of your findings for sargassum management, especially as it relates to offshore harvesting?
So at the moment, we're testing sargassum at different distances from shore. And what we really want to do is assess not only what is there, but if there's any changes. And as the mats approach the shore, where does the species live? Where is the highest which distance from shore has the highest associated biodiversity, and maybe harvesting wouldn't be advised there. So we're looking at tracking the changes, and if we can really identify how the map changes as it approaches the coastline and put a distance on it, and we can actually inform best harvest practices, and that's the direction we're hoping to go. So far, we've only been able to go between 0 to about 2,000 meters offshore, and most of our biodiversity, our free swimming fauna, have been observed between, let's say, 800 and 2,000 meters offshore.
Thank you. We have a question coming through for Bethia now. What has the government's role been in outreach and education, if any, and what sectors of society have these campaigns targeted? And this is a question coming from Michelle Shera Pierre.
Okay. Thank you, Michelle, for your question. Like all other governments, when Sargassum came on stream, it took them by surprise and they scrambled into action. However, to date, under the CC4 Fish project, National Sargassum Management Plan has been developed, and this is under review with SERMIs to better it and to get it to where it's supposed to be. Additionally, under the same project, CC4 Fish, some public education and awareness activities were conducted in a few of the communities that were severely impacted. Tools were also provided to the person so that they could take charge of their own cleanup. Unfortunately, this has been met with limited success. Persons still believe that the responsibility for cleanup is the government, and they don't seem very forthcoming in wanting to clean up themselves. Additionally, we are guided by a number of tools from SERMIs, like the management brief and the Outlook bulletin, which helps us predict when Sargassum can come on stream. And so they're making headway. The target persons generally tend to be community members and primarily fisher folk. A few of the tourism associations have been involved. Unfortunately, the Ministry of Health has not been seen as a very valuable stakeholder, and we're hoping that these things can change. The point of one of the aims of my research is to allow community members to see that they're the ones that live with the vulnerability, and as a result, they're the ones that can tell the story, and they're the ones that can demand a certain level of redress, and at the same time be responsible for managing Sargassum in their own community. So thank you. I hope that answers your question.
Thank you, Bethia. There's another anonymous question come through for you. What physical features or social aspects along St. Lucia's East Coast make the areas that you selected for your study especially vulnerable to Sargassum effects as opposed to other locations? And I'm going to, as the chair, ask you to answer the first aspect, which is the social aspects, and ask Karima to perhaps step in on the physical vulnerability. Thank you.
Sure, with pleasure. Okay, because a PhD is resource bound and time bound, we had to accomplish this in 3 to 4 years. However, there are 11 sites along the east coast of St. Lucia that are currently affected by Sargassum. This information was provided to me by Mr. Anthony Philogence, which happens to be the head of the Sargassum Task Force. Whether it's operational or not is another story. However, we visited those 11 sites. We took photographs and videos. We did a site profile, and we did a historical profile as well. And based on that, a matrix was created and those sites were selected. The social aspect would be most of these places are direct, the communities are in very close proximity. Additionally, when the Sargassum does come, it doesn't dissipate or settle like others, it becomes trapped. So the smell is a very big problem, being a nuisance to persons. Additionally, those communities happen to be fishing communities, which is the primary livelihood of those persons in that area. And so based on these 3 criteria, along with 2 others that have eluded me at this time, we were able to narrow it down to these 3 being the most severely affected out of the 11.
Thank you very much. Karima, can you weigh in on the physical aspects?
Happy to. So in the mapping that we've done, it's still fairly high level, but from observation, I think that we can say, for example, that impacts on an area where there are cliffs will be different from impacts on an area where there's a beach. That's obvious. To give some examples from Barbados, if we look at Oystons Bay, which is just sort of somewhat sheltered by the southern point of the island, that is a bay that we know has relatively poor circulation. So in that area, more so than sargassum piling up on the beach, you tend to see sargassum decomposing in the water and the water turning brown and having those kinds of impacts. Whereas on the southeast coast of Barbados, on the other side of South Point, where things are more exposed, there are quite a few sort of small pocket beaches that are bound by cliffs or rock outcrops. And in those areas, you tend to get sargassum piling up quite thick in the sort of concavity of the pocket beach. And those are difficult to access from the land for clearing or responding in general. So you see these differences in terms of the type and the orientation of your beach in terms or of the shoreline in terms of whether or not it's a wide beach, a narrow beach, whether or not they're cliffs, or in terms of the characteristics of the area in terms of circulation are all going to affect the type of impact that you're seeing, whether it's sargassum piled up in deep or whether you're dealing more with decomposing in the water column. And this is something that I think it would be interesting to do more work on in terms of mapping. So for example, in Barbados, we have mapping characterizing the shoreline in terms of whether it's eroding beach, accreting beach, whether it's cliff, to overlay that with the sargassum exposure and maybe collect some additional data on the types of impacts that we're seeing at different locations would allow us to perhaps draw some correlation between, in a more scientific matter than just these types of observations, between the physical characteristics of the beach, of the coastline, and the impacts of sargassum influxes.
Okay, thanks, Karima. We have a question here from Nancy Brown-Peterson to Mariana. First, a compliment, really great use of mapping to show the various stages of sargassum inundation. Has there been any water quality work done regarding the brown tide aspect and how it affects fish and fishers?
Thank you so much. Yes, actually, those two hotspots in La Valguera are also study sites for other studies conducted by the University of Puerto Rico at Mayagüez. And there are some students working on water quality that I can share with you their contact information. And also there's some research on mangrove productivity as well. Related to fisheries, I haven't seen research done so far on that, but I know who to ask to see if there is something. Yes, so basically, and that part of water quality that I did, I used different water quality indexes to be able to pick up the spectral signature of sargassum brown tide. So that is something that that we were able to do, and hopefully we had to test it on other areas and see if it's able to pick it up as well. So yeah.
Okay, thank you. And just one more before I hand over to Hazel, from Michelle. Mariana, can this monitoring be conducted by local managers, for example, coastal zone management or MPA managers, to forecast next year's accumulation calendar at priority sites?
Well, yeah, so this product allows you to see to see what has been, where sargassum has accumulated in the past. And that is because we're using Sentinel-2 satellite data, and that is an archive. It's not something that is in real time, so it gives you, towards back in time. Basically, it's the information to be able to forecast. That will be other types of methodology and other tools that I'm not currently working on. But there are other researchers that are doing it. So maybe it could be used in conjunction with other types of methods in order to see where are the areas that sargassum gets accumulated the most, and where are those hotspots that are maybe more important to act more immediately. And then see if with other tools, see if there is indeed sargassum coming on the way. And another thing related to the use by managers, right now the methodology is in a JavaScript code that is within Google Earth Engine. And although it's really easy for someone that knows the code to apply it to other area, we will have to explore if in the future a manager or other person could be able to adjust just simple things and be able to use it for their area. But that is something very important that I think we can for sure take it into consideration as a product for the end. So thank you.
Yes, thank you. Oh, and Michelle also had one last question for you. How do you plan to communicate these important results to the client, I guess the MPA manager or homeowners association? And how do you integrate your findings into sargassum response plans across the island?
Yes, that is a very important aspect, and because this research is part of a bigger research that already incorporates the collaboration with stakeholders and a lot of different meetings for the assessment of vulnerability. We are already in conversations with local and federal agencies and also the communities, the Palmas del Mar Homeowners Association, for example. So we are already in those conversations, and they are aware, and they can comment. We have an open conversation, but also we will have some specifically some activities, participatory activities for them to be able to participate and let us know how we can adapt or incorporate things in order to make it more useful for them. So that is incorporated. We're in those conversations, and the overall goal is to make a product that is useful for them so if anyone has any good recommendations on how to improve that, and I'm happy to hear it. So thank you. Yeah.
Okay, thanks, Mariana. Over to you, Hazel.
Thank you indeed. Thank you, Mariana. You're getting all the attention at the moment, because as people ask questions, so more questions arise. And I certainly had one which I think you've actually already answered, but how can we use this across the rest of the Caribbean to perhaps retroactively validate some of our forecasting efforts? In other words, is it something that you can release that other people can use and copy?
Yes, I forgot the name of that term. There's forecast and there's the other term.
Hindcasting.
Yeah, hindcasting. Thank you.
Yep.
So yeah, it could be used for hindcasting completely. And I think one of the biggest reasons why I decided to use Google Earth Engine, it was because it allows you to quickly change the data that you're using. For example, I could easily put it in Barbados, the study area, change it, and quickly apply the code to that area. obviously the training data that I use is for Puerto Rico, but if we feed if we add some new data, we can quickly adjust it and see how valid it is with an accuracy assessment. So yeah, it's something that it can be done. I am working on it, and as soon as I can publish it and put it out there, I will be happy to collaborate with anyone who wants to use it in your area. So yeah, that would be amazing.
Oh, that's great. It has, I mean, I see it as having enormous application to all sorts of studies, particularly when you're looking at the brown tide and there's people studying the impacts on reefs and habitats and so on, to be able to go back using your stuff to look at what the exposure was, if you like. No, really, really nice work.
Thank you.
I've got some difficult questions coming through from Don. I think he probably posted them after watching the talks. I have one from Christy here. Sorry, for Christy, from Don Johnson. Do you have any plans to include the stuff these animals feed on while transiting the ocean? I guess I am wondering if edible coatings might be differently picked up in different environmental backgrounds and set a foundation for what species of fauna are attracted and remain with the Sargassum floats.
Hi, Don. So I don't have any plans to feed the animals I am observing right now, but in terms of the different backgrounds they may come from, there are plans to do some hang casting to determine exactly where the sargassum mat came from and if that determines what species differences we see, but that hasn't been done at the moment.
Thank you. Yes, I think that's going to be a really interesting aspect of your study. I have a difficult question for Karima from the same Don. I guess that's why he hasn't showed his face. From a physical oceanography point of view, I get the coastal morphology and inclusion in your future investigations, but would you consider the offshore approaches to your islands? Flow tends to go around islands. Local winds, waves, and offshore topography make landings happen. Have you considered including these things?
Thank you for that question. And I think the way I'll answer that is that I would love to. Let me unpack some stuff if I can try to do that. So the way that we have tried to assess exposure for the time being is really using historic data extracted from Google Earth. So that's kind of more empirical. And I guess what we're essentially saying is that based on what has happened in the past, we are kind of extrapolating the probability to some extent of that happening in the future in terms of exposure to influxes versus a numerical modeling predictive approach, which would be based on the physical processes that are occurring that are bringing the sargassum to our shores. And I think the limitation on looking at that in terms of understanding exposure and the frequency and probability of landings is really data. So for Barbados, we have quite extensive data. Barbados actually has an island-wide wave climate model that was done under some work a few years ago under the Coastal Risk Assessment and Management Program. But that is not something that is going to commonly exist across the region, especially at an island-wide or a country-wide level. So I think that would be a limitation on doing significant modeling work in terms of predicting landings based on the physical processes. What we could possibly do is something and maybe a bit more, I don't know what word to use. I will tentatively put forward synoptic, where if we have some general understanding of the directions of flow, directions of currents, we may be able to sort of incorporate that into the mapping and look to see if there are any general trends that we observe that may be useful. But I think that the main limitation would be in the available data to do numerical predictive modeling. I hope that answers the question. And I'll take the opportunity to sort of just make the point as well. I see there's a question about deploying equipment to entrap sargassum before it strands. Those same processes that are bringing the sargassum to our shores and the conditions associated with them also affect that feasibility. Those things vary in space and time. So the feasibility of that type of equipment approach to trap sargassum before it lands varies in space accordingly. I would feel much more comfortable with equipment trying to collect sargassum offshore of Belize at the much more sheltered western side of the Caribbean behind a large barrier reef than I would feel in the open Atlantic offshore of Barbados with that same equipment. So that's my take. I hope that was useful. Thank you.
Thank you, Karima. Very interesting thoughts. There was a question here from Alejandro Acosta. I believe this may be directed at Christy or anyone else who wants to take the question. What is the impact and magnitude of sargassum in fish recruitment on natural reefs?
Hi, Alejandro. So while my project is not focused on identifying the impact or the impact associated with MAX and habitat recruitment, I did read there was a study in Mexico who was trying to do something similar, and they're actually trying to determine what the role the MAX play with inshore and offshore reefs. But I haven't seen any outputs from the study, and in my opinion, I think this would vary as well, depending on the benthic environment where the mat is in. It depends on the thickness of the mat composition. It depends on a lot of stuff associated with the mat, and I don't know what that impact is very offhand, but it is a very important question.
Yeah, great research topic, yeah. Alejandro. You need to put a student on it. That's certainly a research gap. And there was another one for Christy back a while ago, asked by Mark Peterson. And he said, we saw changes in bryozoan mats in the, I guess it was North Gulf of Mexico study. That is in diversity differences onshore, offshore. So I guess he's asking if you've seen any differences onshore, offshore, particularly in bryozoan mats.
I haven't seen any differences as yet, but I do believe Mark said he sent a paper which would be, I guess it's around the bryozoans as well. He sent it to you, Hazel, so you can send it to me after.
But you have seen differences in quite a lot of the clinging fauna in particular between onshore, offshore, as you presented earlier.
The clinging fauna, yes, but the epiphytic fauna, which are the bryozoans, not as yet. But this is only 2 months of data, so with more data, that may change.
It would actually be nice to throw that question back at Mark, if he's on the call, to ask him why there might be changes. Why would you expect changes in epifauna between nearshore and offshore? Don't know if we're allowed to ask the general audience questions. Maybe they don't have a microphone, so sorry about that one.
Okay, maybe. Yes, I am. Another question from Mariana coming in. Okay, yes, so Mariana, did you attempt to use the AFAI, which is the Algae Floating Index, and the NFAI indices in your methodology, sorry? And these would be, indices used by the Sargassum Watch System, as well as CLS in the preparation of their products that we use, in forecasting.
Yes, great question. So I use a combination of different indices, of vegetation indexes and water quality indexes. Originally, I intended to use the floating algae index. However, the way it requires certain level of correction of the imagery that that I can't do in Google Earth Engine right now. Therefore, that specific index I was not able to use in the processing. However, I use other as NDVI, plenty others. Yeah, have plenty others of vegetation indexes. However, we are planning on doing a separate analysis and processing the same area using other program, not Google Earth Engine, and processing it with the floating algae index, and then comparing the results. So to be able to see how well this new product does in the coastal area related to in comparison with the floating algae index. So we are going to do it. We have it. Yeah, we have it in plans.
That's good to hear. I will be very interested in those results for sure. Okay. Oh, Mark has actually answered in the Q&A. Mark was saying that we felt that it was inverts and fishes offshore differed than inshore assemblages, quite distinct. We bagged, cleaned Bryozone maps in bags and set them up along a transect and found many differences. Okay. Christy, any reflections on that? Or I guess Christy and Mark needs to have a little coffee after.
I was thinking the same thing, Shelley, because
Yeah.
now I want to ask Mark like how far his, what his transit distances were, and I just want to talk to him about his study, but I can contact him after.
Okay, I'm going to plug for the WOVA app. I believe there's a way that you can do a hookup and a meetup in the app as well too. So yes, you can feel free. He says it's all in the paper. Yes, yes, yes. Okay.
But I think you should talk to him anyway.
Yeah. Okay, another question from Michelle to Bethia. What has been the effectiveness of local communities with accumulation so far?
Accumulation of the Sargassum?
I believe that's what she's referring to, yes.
Okay, we actually have one success story coming out of the community of Denry, which I'm actually studying. A young man, Johannan Doujon, has actually been able to use the Sargassum to create a plant tonic, and he's created his own company. He's now the CEO of his own company. It exports to 3 countries in the Caribbean and 1 international country. It's called Algas, and he's been able to use the support of the community as collectors, baggers, truckers, and grow his business. He first started maybe about 8 years ago, and right now his products are on the shelf and in Caribbean islands, and persons are attesting to that. You also have some persons who utilize it as composting purposes. Some local farmers who collect it, wash it, dry, allow it to lose the salt, and then apply it to their crops. However, you have some with aversions because they've heard of the possibility of arsenic and that sort of thing. So they're requiring further information. And you have the majority of them who have to remain there, and persons have even become indifferent, telling you that that's just an act of God. There's nothing we can do about it. And whether we know it's coming or not, we cannot change that. And so they've sort of come to accept it. So you have the 2 sides of the spectrum. Those who believe that we need more information to see how we can make this something good and get money out of it for job creation. And you have others who say, just let it remain. It's been coming for years. We've gotten used to it. We wear our masks anyway. We close our windows and we live with it as it is.
Okay. Thanks, Bethia. Okay, to Karima. We noticed you mentioned that you were at the COP. So we have quite an interesting question here as it relates to the improvement to access to coverage of aerial images. Do you think this will be a priority given the outcomes of COP26 and the availability of funding to support climate adaptation?
That's a fun one. Thank you for that. Honestly, I'm not sure what to say. To be honest, the discussions that I've been a part of are not at that level of detail in terms of the specifics of what climate finance should support or what types of adaptation priorities there should be. The discussions have been more general in terms of things like amounts and sources of climate finance, and well as issues around adaptation in terms of how we track progress on adaptation and methodologies for that. Those are the kinds of discussions that I at least have been a part of. And I haven't heard anything nearly so specific in terms of what technologies or what activities may be prioritized. That said, I have not had nearly as much opportunity as I would like to sort of wander around all of the expos and the presentations to see what other people are up to. If I get the opportunity to do that this week and I see anything of interest, I will be happy to report back.
Thank you. Thank you. Good response. Okay. And let's just check the chat box and other questions. Okay, now that Karima is still on the microphone, what are your recommendations though for applying the template to other countries in the region? I know you've started with the pilot with the 5, yeah, but how easily can it be replicated and perhaps scaled up?
The overall process is really quite straightforward. We're really just applying some basic methodologies for mapping exposure and mapping assets and overlaying those so that we have that spatial understanding of what assets are exposed to the influxes. So at that general level, it's quite straightforward to apply elsewhere. I think where the detail will come in then is what data is available for each location, both in terms of the Sargassum influx data. We found in the Google Earth images that we were working with that, I mean, simply as a matter of the smaller islands in the Eastern Caribbean, some of them, there were far less images than there are for other locations or something like that. As simple as that is a bit of a limitation. Some locations may have other sources of data on historic influxes that can be used to develop that kind of empirical assessment of exposure. And then some places may have a greater level of spatial planning already, mapping their assets in terms of tourism or fisheries or other categories of interest, whereas others may have less. So I think it would come down to the details of available data, but the general principle of overlaying some information about exposure with some information about assets, and then examining that to get an understanding of what the spatial trends are and looking at how that can be used to inform planning and policy would be quite straightforward to apply anywhere that there was interest.
Okay, very interesting.
And I think would be very useful for informing the planning process and prioritizing resources. Yes, thank you.
You're welcome. Yes, Hazel, I saw you had a couple of questions there.
Yes, I have one for Christy. I think it's a very interesting question that a lot of people from the Gulf of Mexico and Bermuda are going to be keen on is, does the biodiversity that you've looked at so far in the Caribbean or the tropical Sargassum differ from the biodiversity that's been reported for the Sargasso Sea?
Thanks, Hazel. The biodiversity that we've observed so far, I can find in literature from the Sargasso Sea, but the biodiversity Sargasso Sea, and there's lots of literature there, is like we would find 2 crabs and they'll find 4 to 6 crabs. Like we have a reduced biodiversity so far. It could be the fact that I have just started sampling, but I haven't found anything within our data set that hasn't been reported to date in the Sargasso Sea. However, I say that noting that I have like a few more species to identify. I haven't quite identified them yet. So if they're not in the reported Sargasso Sea literature, then maybe there'll be slight differences, but I can't report on that as yet because they haven't been identified.
Okay, thank you. I mean, I think it's very fascinating. Certainly the Sargassum in the tropical Atlantic, the bloom there is much younger than the bloom in the Sargasso Sea, and maybe it will take decades to build up the same biodiversity. I think it's an interesting question. So I look forward to another year of your field work and see what shows up. Thank you.
Yes, I see we have a hand up from an attendee, Gracilia. Do you have a question to pose? Okay, she's been permitted to speak.
Hoping to hear from you, Graciela.
Hello?
Yes, we can hear you now. Please go ahead.
Would need for you to repeat the question, please.
Oh, sorry, your hand was up, so I assumed you wanted to answer a question live.
Ah, no, sorry about that.
Okay, you were just waving then, that's fine. That's okay. But yes, thanks for joining this session and thanks for supporting our panelists this afternoon. Bethia, how willing were the invisible, as you refer to them, to interact and share their experiences with you? Did you experience any, I guess, some community members that weren't willing to share, or I guess not open to speaking to someone, I wouldn't say outsider, but yeah, so what was your experiences like in the field?
Yes, thank you, Dr. Cox, for that question. Actually, it's quite interesting. I am from one of the communities, the community of Miku. So this one, in terms of familiarity, was fairly simple, although it can work in your disadvantage too by being too familiar. Additionally, I have worked with the Department of Sustainable Development, so I have been engaged in fishery stakeholder work before. So I'm sort of, I wouldn't say known, but I'm sort of familiar to a few of the persons. Additionally, the groundwork was very important. I would go to the communities. I would let them know in a few weeks I'll be coming in to speak to them, do some data collection. While we collect the data, I let them know that there is this upcoming meeting. I'm speaking to the fishermen as well. I try to get them excited. I let them know that we're going to be making this video and you're going to get the chance to say it how it is. And you can speak in Patois, which is our second language if you wish, and just express your views. Additionally, we always coax them with a little refreshment. That always helps. A little chicken and drink, and we try to work with their schedule. COVID did stir up its head. We had some challenges with elections around the same time, and then the hurricane, but we changed the dates a few times. But as the word got around and persons, you naturally get a few who'd say, I'm not interested in that. There's nothing you can do. Persons have come to me before and I don't wish to speak to you. But we used our charm, and we got a few for the Prale community meetings. We had 13. For Dennery, we had 17. And for Miku, we had 22. And so we think amidst everything that was happening, this was a good representation. And surprisingly, the persons were very knowledgeable about Sargassum, very willing to participate, very willing to share their stories. And yes, it has been something to look forward to. I'm excited about the videos that are coming up. They've done all their groundwork. They've written their script. They've taken their videos and their photographs, and we're now in the process of getting a videographer to collate everything. And as soon as that becomes available and it's possible to share, we will do so with the scientific community. Thank you.
Okay, thanks, Bethia. And we have another question from Anonymous. How have pelagic fisheries been responding to the inundation? Is increased sargassum enhancing certain populations? Are landings increasing? Or are the obstacles that sargassum presents to fishers negatively affecting pelagic fisheries and perhaps reducing fishing effort and landings? Quite an interesting question, and it has intrigued Hazel and I in the past, and some work that we've been doing in the region, especially looking at the decrease in landings. In Barbados, for example, where flying fish accounted for about 60% of landings. I recently saw the chief fisheries officer presenting that the landings are closer to 30%. So quite significant reduction in the landings. But of course, with anything in terms of coping and adaptation, people are now targeting almaco jacks, or amberfish as it's locally called. And, there is a market now where people are now asking for the fish. It's quite tasty and soft. And we've seen some chefs as well getting involved in preparing it in creative ways. So your question here, are landings increasing? No, they're decreasing, also decreasing for dolphin fish. Hazel and I and our colleagues, we've wrote a report on, the relationship between sargassum landings and the decrease of flying fish and dolphin fish. We can share that with you. And finally, Hazel, I don't know if you want to take this last question. I can ask it. The obstacle that sargassum presents to fishers negatively affecting pelagic fisheries and reducing fishing effort. I believe,
Shelley. We think the questions seem to be coming at us as well as the other panelists. So I'm happy to share the many years of work that we've done with communities. And I'd like to hear actually from Bethia whether she's hearing the same thing in St. Lucia. But yes, it depends on the fishery, I think is the best answer. And as Shelley said, our flying fish fishery in particular has been very badly affected by sargassum. But I suspect that the species has not. It's just the fishery that has been impacted. Our fishermen use fads, which attract flying fish to lay their eggs, and that's how they catch them. When there's plenty of sargassum around, they don't need to go to fishermen's fads. So there's been a huge reduction in catch. In terms of dolphin fish, probably the numbers of dolphin fish that are caught have increased, and the seasonality of the fishery has expanded through the summer. But that is because they're catching baby dolphin fish under sargassum now. So the baby dolphin fish are now available to the fishery, and they weren't available before. But in terms of weight, the landings are down because the dolphin fish are being caught as babies, and therefore the larger fish that would have been caught later in the year, they only live a year, are no longer available because they're being fished out, or so it seems to us. And then there's some other species like yellow jack that we never saw before, and the amberfish, or the almaco jack that Shelley spoke of, are new species that have come into our reef fishery. And fishermen also speak of more availability of certain species like barracuda since sargassum arrived.
Yeah.
So it's adaptation, changing market. Perhaps you would like to add to that, Becky, on the people you've spoken to in the communities.
Yes, Professor Oxenford, it's a similar situation here. For us, it's not so much the flying fish, but a species that is locally known as balawu. It's a very slender fish with a pointy nose. And we have the almo jacks in abundance. The fishes, however, have spoken about not going to familiar spots because the sargassum hinders their navigation through the waters. Others have abandoned it altogether and are doing other things like maybe construction or gone into farming. Interestingly, the persons in Prale are not as adversely impacted because there is sort of a diversification into sea moss farming. which is very big in Prague. And a concern for them is not so much how it affects their fishing, but how that sargassum in turn will affect the sea moss farms just a little way off from the shore. So interesting conversations, similar things for species size and catch composition, just like you have reported. They have noted that it affects their seagrass beds and their mangroves. There is deep concern that because it's affecting the mangroves, they will not get the baby fish, the nursery fish that then go into make the bigger fish. And so these are some of the concerns they have right now.
Okay, thanks, Bethia. And I'm sure we've heard from, other fishers across the Caribbean who are also having similar experiences. Okay, well, it is 4 minutes to the top of the hour where our session ends. Right now, we don't have any more questions being posed. I want to thank our panelists for, very good presentations, responding to the questions so well, taking part in the discussion. Of course, for those attendees, if you haven't watched their presentations before, please take a look to find out more. And as well, you can contact them for further information. I am going to hand over I have to jump off at this point. I'm going to hand over to Hazel to do the final wrap up. But thank you, it was a pleasure.
Yes, thank you very much. And I must say congratulations to GCFI, because I found these panel discussions really interesting. So rather than just listening to presentations and then everybody running over and not having any time for questions, it's actually been a really, really nice discussion. And thank you to the audience for all of your very interesting questions, which I hope will continue into the future on sargassum and related issues. So indeed, thank you panelists for your fantastic short presentations, and as Shelley said, really nice longer presentations that are available for the audience to watch in their own time. And I really look forward to seeing the results of your work in the literature and to actually using and applying your results as we go forward. So thank you very much, everybody. And time to hand back over to Fadila, who's doing a phenomenal job of running this meeting.
Thank you, Hazel. Thank you, Hazel, and thank you, Shelley, for moderating such a great session. It's really our honor to have two of the leading experts on sargassum be part of our GCFI panel discussion. And again, thank you to all the panelists today. And as I would just like to reinforce what both Hazel and Shelley said, even though the panel discussion is now complete, we encourage you to please look at the presentations if you haven't already, and continue to engage with the speakers and panelists and all the different attendees. So thank you again for this wonderful panel discussion related to sargassum. And now we are going to transition to our next panel discussion, which will be related to marine protected areas. So thank you again. And I would invite Emma, our moderator for the next session, to join us, and then we can get our presenters ready. So I can see quite a few.
Perfect. I can see that we have Ian, Palayo, Lee, and Julian, and let's see, that Leslie. There we go. That's looking good. All right. I think we are right on time then to start. Let's go ahead and get going with our MPA session. Good afternoon, everyone. Welcome to the GCFI74 technical session on Marine Protected Area Science and Management. I'm Emma Doyle with the Gulf and Caribbean Fisheries Institute. I serve as coordinator of MPA Connect, the Learning Network, which is a partnership initiative between GCFI, NOAA's Coral Reef Conservation Program, and 32 priority coral reef MPAs in 11 Caribbean countries and territories. Now, the MPA technical session is traditionally part, no, let's save that to come to later, Fadila, thanks. The MPA technical session is traditionally part of every GCFI meeting. I first helped to moderate the session in 2009 in Cumaná, Venezuela, and I'm pleased to bring you this year's first ever fully virtual GCFI MPA session. Now remember, you can view the presentations in full on the WOVA app, and today we feature summaries by the presenters, followed by discussion with the audience and the panelists. We're going to look at MPAs from several different viewpoints this afternoon, and we're going to cover 4 main topics that were raised by the presenters this year. Now, I'm proposing that we have some audience discussion after each topic. So first, we've got 2 presentations on the core management need for reliable, long-term, unrestricted, sustainable MPA financing. Then there's the management challenge of ensuring that dive tourism is sustainable. There's the need for stakeholder engagement and consensus building in MPA planning, establishment and management. And we'll look at the important topic of collaboration for a data sharing gateway and for effective protection of species and places. So our panelists are coming to you from all corners of the region, from the Dutch Caribbean, the Bay Islands of Honduras, work in Bermuda, the US National Marine Sanctuaries, and from 2 regional initiatives. Now, we have a great audience with us today, so thank you for joining us. We aim to have an interesting and a practical and an energetic session with some productive discussion. So please join in, add your comments in the chat box and or in the Q&A box, or raise your hand to contribute. Please let us know how your MPA is doing or what you've learned, or comment on cross-cutting issues that you see between the presentations, because with MPA management, everything interconnects. The discussion isn't just between the panelists, but with all of you in the audience too. We speak Spanish, English, Dutch here this afternoon, if not more. So feel free to speak in your favorite language. And remember, there's simultaneous translation between English and Spanish in the translation button. All right, with that, I would like to invite Arjen de Roon from WWF, the Netherlands, to tell us about sustainable financing in part of the Dutch Caribbean. I'll then present directly after Arjen, and then let's take some questions and comments on and discussion on MPA sustainable financing. Okay, Arjen.
Well, good afternoon. Wait, let me figure out how to make the screen smaller. There we are. Well, thank you very much for having me and allowing me to speak on a project that we helped run on the Dutch Caribbean, on the Windward Islands, Saba, Sint Maarten, and Sint Eustatius. I understand that I only have 3 minutes to give a summary. So I'm going to dive right into the findings. I hope everybody managed to have a look at the presentation that was put online, I think, the 2nd of November already. What we found doing that project is that, of course, funding for MPAs has been and will probably continue to be a difficult problem to solve. Knowing that the biodiversity is mostly out of sight of the general public and the policy makers, it's very difficult to make the case for financing of that underwater world. However, for the Dutch Caribbean, or part of the Dutch Caribbean, we did start making that case. What we learned from the project that we did is that as a MPA, it's quite important to know what you're spending your money on and what you can achieve with the limited funds that you have available. That might be an open door, so to speak, but actually getting your accounting in order was posing to be quite an important part of the sustainable financing project. We also learned that parks are not separate entities and that you when looking for funding, you need to consider the position of, for instance, governments who are also strapped for money and who often have a very different idea of what a park authority should be doing compared to what the park authorities themselves think should be done. So it's very clear to, or very important to have the responsibilities clear between the two parties. Otherwise, you will never agree on what needs to be funded and what won't. So you also have to show what additional funding can achieve on the ground. So if you do get additional funding, what is the impact that you're going to make with it? And which problems will it solve? And how does it benefit the various stakeholders? If you do not communicate or explain this to the people that you work with or your potential funders, they will not understand the urgency to invest in the MPA. We do know now which local opportunities are available on the islands that we worked on, and we actually made a plan to start working on that. And that ranges from a slew of different funding sources, but that requires a proper analysis, which we did within this project. And maybe more importantly, we know the amount of money you could leverage through local means. And we found that the amount of money that you can raise locally is often, or in some cases, not enough to fill the financing gap. So you have to look at other avenues to getting funding. And this is actually something that I very much like some feedback on from the group, what options are out there. And you have to be diverse, just like your coral reefs. The more diverse your funding streams, the more solid you are when pandemics hit, when the climate starts throwing storms your way and tourism stops. So it is very beneficial to spread the deck, so to speak. And any solution takes effort, work, people to do it. Actually, I'm advocating here to consider recruiting a fundraiser as a park, or work with a couple of parks together and recruit a fundraiser, because in the end they will pay themselves back. But there is no silver bullet, and that is the main conclusion. So you also have to look at outside of your own sphere of influence locally, and that is actually one of the take-home messages, take-home questions actually. So what can you do on a regional level, and what can you do with a regional trust fund or emergency climate funding, or those kinds of solutions, or insurance mechanisms. But the regional aspects, I think, are more within Emma's area of expertise. So with this, I would like to leave you with Emma.
Okay, thank you, Arjen. Please, in the audience, perfect. We've got one question coming in. Please add your comments as you think of them. I'll go ahead then, and I'd like, before we launch into any discussion though, I would like to sum up 10 years of capacity building for sustainable financing by MPA Connect, with about 40 participants at 2 regional workshops, 14 financing projects, 6 financing experts we've worked with, a couple of amazing financing mentors in our network. Has there been any progress? Well, by about the halfway point in our 10-year history, we found that fewer of the MPA Connect sites had little or no reliable funding than they'd had 5 years earlier. But there wasn't a lot of progress still in generating long-term funding by implementing a sustainable financing plan. Another 5 years later, we hope to measure capacity again. If you pushed me to make an estimate, I'd say that maybe about 1/3 of our MPA Connect sites are making really good progress in generating sustainable financing now. I think as a network, we're on the right path to make a difference in terms of site level capacity for MPA financing. So I'd like to just reflect quickly on 10 lessons we've learned on what our MPA managers tell us is their #1 need for capacity building. So it all started with a regional workshop in 2011 to share about successful financing strategies. This then prompted our first site level financing exchange as a follow-up activity, but that very quickly showed, lesson #1, that there's more to generating funds for an MPA than simply replicating an approach that's been proven successful elsewhere. Now, that exchange also revealed that MPA budgeting processes needed attention before we could work out the financing gap that we needed to close. MPA Connect has now developed a good MPA budget template, but lesson 2, like Arjen said, is that there's no avoiding the intensive task of calculating the financing needs of your own MPA. Now, if you go to Arjen's presentation, you'll see he sets out the process that MPA Connect has also followed. We've worked with the Eco2Fin model. We've worked with databases like the BioFin catalogue of financing mechanisms. We've screened our mechanisms to prioritize them so that you have unique and creative solutions that are tailored to your MPA. And that's lesson 3. Now, lessons 4 and 5 are that financing can be hard work. Technical assistance from our experts at Wolf's company, who we've worked with, has been really important. And implementing even a well thought out idea still takes a lot of time and effort to put it into practice properly. So patience is a virtue. Lesson 6 is that financing mentorship from peers has been amazing. You know, we trust each other and we get it as different managers. We just need more time and some more support so that we can get together more to work on our finances. Now, the next lessons concern remaining challenges. MPA governance type can make a difference. It can make life easier or harder when it comes to financing. Innovative mechanisms are growing in profile, but there are some barriers to overcome first. And we need to demystify some of the newer concepts, you know, like blue economy, blended finance, some things you'll hear and need to know and be able to work with. Now, I always say that in MPA management, everything comes down to effective communications. So the second last lesson is that this is especially true in raising financing or tackling barriers with specific target audiences. Like Arjen said, communicating the impact. Now, lastly, I'm seeing some meaningful financing partnerships being forged upon principles of transparency so that funding partners of whatever type contribute funding directly to the MPA's management objectives. So now we just hope to speed up the work and extend our work on this important topic. Okay, that's it from me. That's no mean feat to summarize in 3 minutes, by the way. Now, there is a question I'm seeing here. Arjen, can you elaborate on some of the local fundraising strategies that are being implemented? That's coming from Caroline Oliver, believe in Tide Belize.
There's actually, for the Dutch Caribbean, quite a few that are currently being implemented. Before I go into that, just a quick note that I didn't put that on the slide, but the parks did most of the work. Wolf's company did most of the work with the parks, and DCNA facilitated everything. So as WWF, we support and we fund, but the work was done by the partners. So I wanted to flag that because I'm telling the story, but they did the work. So I want to highlight that. And then with regards to the funding or financing options, there are a slew of different things that came out of the assessment, ranging from strengthening the DCNA Trust Fund for the Dutch Caribbean. I don't know if everybody knows, but there is a trust fund hosted by the Dutch Caribbean Nature Alliance, which has, I don't know, 18 million in it or something. And they disperse funding to the 6 parks in the Dutch Caribbean. But that's not enough to run the parks. It's a little bit of core funding that they get every year. So that is actually something that we are pursuing SWWF within the Netherlands to try and get the Dutch government to fill that trust fund so that that disbursement every year goes up for all the 6 parks. And we're getting there, but that has been like a 10-year struggle. But that's one option. Then obviously there is the fee structure where tourists pay for either using the ocean or using the park, or they pay tourism fee when entering the island. And that is for most of the parks the main source of income aside from subsidy from the local government. But that is very fragile in the sense that during COVID, as with many of you, the income just collapsed. And luckily we have a Dutch government that kind of had some emergency funding available to cover that gap, or at least part of it. But that's the current status. And the changes or the new solutions going forward for Sint Maarten, for instance, they were looking at liaising with the local government to get the cruise sector to pay a fee. And it doesn't have to be a large fee because there is more than 1 million tourists that come there. So if you leverage a fee of 2, 3, 4 euros, you will also already have quite a big chunk of your funding in. But this is a kind of a how do you say that? It's not a new system. It's just that they needed the support to actually get that going. And on Statia or St. Eustatius, they are currently also talking with the local government about leveraging a visitor fee for everyone who enters the island, of which half will go to the government and half will go to the park. On Seba, they are working on a marine lab that they can make available to visiting researchers. And they hope to generate not much, but a little bit of funding from there. And there's more, there's donations, there's grants, there's the advocacy work that we help them with to get structural funding from the Netherlands. But I understand that not all the countries in the Caribbean have that luxury. And we're looking into a climate emergency response fund. All those kinds of things are on the agenda. But if you look at the presentation that I gave earlier, there's a list for Sint Maarten of all the options that were presented by Wolf's company in collaboration with the stakeholders. And it's a lot. The only problem is that it is also never enough to get the amount of funding you need to actually protect nature properly, or at least for the smaller islands. On other islands, that's a different case. On Bonaire, for instance, the money that comes in through user fees basically covers everything they need. So it changes. It varies per location.
Okay, great. Thank you, Arjen. Yeah, I think one of the key points is that there are so many different financing mechanisms, and there are, yeah, Arjen, you've mentioned a lot of them. Other ones that come to mind in our network, crowdsourcing, online marketing, MPA enterprises with profit sharing agreements. Cost effectiveness is quite a valid part of sustainable financing as well, and seeking cost efficiencies. For example, by new mechanisms like online fee payment, although it might not increase the funding you bring in, it can make things more efficient and have other benefits. So that's also a valid approach. The point being that the mix of financing strategies really need to be unique to each site and to the context that they will operate within. I can see there's a question from Leslie. And for MPA Connect, I'm also similarly kind of representing the experience that our network of 32 MPAs has had. And there's quite a few of the MPA managers in the network who are actually online as attendees. And I'd welcome them to jump in at any point. As Alejandro is raising, political willingness is important. And yes, overcoming barriers, their political barriers, social barriers, legal barriers. That's important in terms of some of the innovative mechanisms for financing. So the opportunity perhaps is typically in MPA Connect, it's our MPAs that have direct interactions with funders and with the decision makers, the budget holders, to answer Leslie. There's perhaps potential to do stuff at network level, but that we're gradually working on. Dana asked, how are we going to can I elaborate on how we track increased capacity for sustainable financing? Well, we do an MPA management capacity assessment roughly every 5 years, which is an in-depth look with the managers at their existing self-assessed but guided assessment of capacity for 20 different elements of MPA management capacity. Financing is one of those. And so we'll look carefully. We hope again very soon we'll start to look at what are the needs, what's the current status, to help target our capacity building efforts further. Alfonso, what about lessons learned and tangible products derived from the, measuring effectiveness? As a network, we look to measure effectiveness on the depth of connections and how well connected people are, the fact that help is only one step away, so that we have mentorship to help you with getting to grips with budgeting, or to help share experience on particular mechanisms. Okay. Panel, are there any other comments from panelists? Would any of our MPA manager attendees like to comment? Communications is a critical part of this. So knowledge transfer to governments, highlighting exactly what the budget need is. The Turks and Caicos have done an interesting job of looking at the value that their marine protected areas contribute to nature tourism, for example, and looking at the budget that they receive to manage those natural areas, looking at the gap between those two, and trying to make a really good case for the fact that for a small investment, huge value of nature is protected. So there's a very important communications message in that. And we've also worked with tools like the message box tool to help guide the development of messaging in support of financing.
I'd like to, if I may, go back to Alejandro's question about the political will, because he's exactly right that in a lot of cases that is a severe bottleneck. And of course, communication is part of it. But I'd like to mention an example that they did on St. Eustatius with Stenapa, where they ran into the same problem. But then they started talking about, okay, what can we do that benefits both the park and both the government? And the moment they found a solution like the visitor fee for people leaving the island, it became interesting for the local government to pursue this, because if they would make that happen, they would get half the funding and Stenapa would get half the funding. So it would be beneficial to both. So that's completely unrelated to making the case that it is necessary for nature. It's just a simple matter of, can the government make money with something? And it's never that simple. I mean, don't get me wrong, but it's an example where they started the discussions with the local government and they came to such a solution. So it's worthwhile to have that discussion with your stakeholders, I think, and also consider their point of view in that sense.
Perhaps I'm seeing that too in terms of transparency, that some of the MPAs who've really developed they've developed MPA budgets based on their management objectives to achieve their mandated objectives and achieve, the good for national, cultural, natural heritage. Where MPAs have done that, rather than developing budgets that are just based on project deliverables, or that are constantly crisis level budgets, where there's transparency and they can communicate that to the conservation trust fund managers, to their major donors, then through COVID, we've actually seen that those donors have come to the table and have understood where the critical needs are in achieving the management objectives. And they've shown flexibility, not just to achieve project deliverables, but to really switch to support the MPA management objectives. And now that takes a big leap of faith for an MPA to share its budget with its donors and with important partners. But those who've had confidence in their projections, in their numbers, in their financial planning, have been able to do that and work effectively, and they've benefited from flexibility in donors and increased funding in times of crisis, and hopefully going forward. So rather than constantly making grant proposals, it's a much closer and much more trusting, open process of working together to achieve the financing needed. But that's just one example, and maybe that carries over into impact investment as well, working on blended finance on other solutions. Okay. Shall we come back to?
Do we still have 30 seconds or so? Because I actually have a question for the group, if I may.
Okay.
Does anyone have any experience with regional solutions to financing for individual parks, apart from the MPA Connect work, but a large donor or a large trust fund or whatever kind of solution, or a parametric insurance scheme for the protection of coral reefs or something like that, that they can share something about?
Do we have any takers? Feel free to raise your hand.
Yeah, it's a legitimate question because I'm quite interested to learn more about that, but that might be a discussion for aside.
Okay, we can follow up with that too separately. All right, thank you everyone. If any more thoughts come to mind, then we can continue later. Let's move on, make the most of our time. Let's turn to sustainable tourism. I'd like to invite Pelayo Rico to tell us about his research into scuba diver carrying capacity on Roatan, Honduras. After his presentation, let's take some questions and comments about sustainable tourism management and MPAs. And he is also a student award presenter. Thanks, Pelayo.
Well, hello everyone. Can you hear me okay? What I'm here presenting in this conference is the results from my master thesis, to summarize all the 2 years of studying. And since I was very young, I'm a passionate scuba diver. But when I really take my career into marine biology serious, I started to see scuba diving as two options, like the fun, enjoyable activity that it is, but also that the possible impacts that has on these coral reef environments and all in other marine environments. So what I tried to do was to define a protocol to calculate and control, or to know about the impact that the divers have on the coral reef. So for that, I used the carrying capacity method, that is a calculation that tells us the number of divers allowed in the coral reef that will support the actual state of the coral reef with no degradation. These carrying capacity calculations are made taking into account many factors, as for example, the impact factor, that is the actual contact from the divers with the coral reef. Also, the social factor, that is the physical part, that how many space does the divers occupy in the dive sites, and other factors that in this case there were not significant. I did the study on Roatan, that is one of the Bay Island archipelago from Honduras, I chose this island because most of the tourism of Roatan, that it has over 1 million tourists every year, well, before the break hits in the activity because of COVID. And most of this tourism is diving tourism, especially concentrated in two main areas of the island, that there are Western and West Bay. So as all the Bay Island archipelago are declared as a marine protected area, I saw an opportunity to use this carrying capacity as an adaptive tool for management of the marine protected area in order to control the impacts of the divers and other related activities with scuba diving that take part in this marine protected area. Also, while doing this study, I discovered two additional possible factors or impacts on the coral reef. That one of them was the use of a lionfish spear, that after the results, it was seen as a trade-off between the invasion of the lionfish along the Caribbean, but also the impacts that the divers with the lionfish spear increase when they use it. So it's kind of controversial there, and I think it's we should focus on a good preparation of the divers that actually spear the lionfish. And also I found something that it was on my mind before when I started diving, that the different levels of certification within the diving industry should be in accordance with the experience and with the respect with the coral reef. Actually, the results show it. But even in the higher certification levels, there were some hotspots that some divers make most of the impact. So what I really focused on this part of the level of certification was that most of the main impacts were on low level of certifications, just because most diving at that point is focused on the enjoyable part for the client. After you increase your level of certification, there are more skills such as buoyancy control and trimming posture that you specified on perfection. So you kind of have more control activity, and you avoid the impacts with the coral reef. And that's all from my part.
Okay, muchas gracias, Palayo. So, do we have any questions or comments in terms of responding to carrying capacities and managing dive tourism? Let me see. In the Q&A, Michelle says, good work, Palayo. How do your results compare with other carrying capacity assessments for coral reefs around the world? Can you make a recommendation for a maximum number of scuba divers per hectare or any other estimate of area for Caribbean coral reefs?
So actually, the second question kind of came before doing the project, as can I calculate like a medium or example maximum scuba diver number for each area, for like a general area? And after the project, I found that no, like the carrying capacity protocol is kind of really specific, especially on the coral reefs, because it depends a lot on the morphology and the type of corals that are part of that reef, especially with fragile species. And comparing to other carrying capacity assessments, that most of them are, for example, concentrated in the Red Sea, where also the diving tourism is quite expansive. I think the good state of conservation of this coral reef showed a greater number of divers allowed in Honduras because of the especially that the good conservation of the coral reef. If you compare with other areas that most diving tourism has changed quite a lot the coral reef state, you can see that most of dive sites are already really poor in the state of the coral reef. So that depends between both.
Okay, and linking this back to financing, it's a challenge and a balance to get right, optimizing the number of perhaps user fees that you earn from your divers against potential for damage caused. So I like the fact, and I encourage everyone to have a look at the method that Pelayo used, because I like the way that you established some numbers and then you adjusted them and you factored in behavior and social issues, because that makes it more like a sustainable tourism model rather than carrying capacity for sheep in a paddock or something. So I think that's an interesting approach. Okay, we have some questions in the chat. Martin is asking, similar question, is there a risk assessment matrix of recreational and tourism diver use and impacts being used anywhere that you know of? Risk assessment for divers.
Yeah, actually, I was going through the literature review and I didn't find any. Yeah.
Okay, thanks. It's good to know. Keep looking, Martin. Ian asks, were you able to compare diving impact with, for example, fisheries impact or pollution impact or bleaching impact, for example?
I was not, because especially after COVID outbreak, everything in the master kind of changed in processes and dates, all kind of focused in just 3 months. I was able to go to Honduras, but actually with the island, we wanted to make this kind of the first step to more work in the future, to try to compare with other, not only diving impacts, but other anthropogenic impacts like the fisheries and the polluting and the bleaching, or for example, the new and quite spread over the Bay Island, the Stony coral loss disease.
Okay, yeah, interesting question. All right. Hold any further questions on this topic and we can always come back if you think of anything, but let's move on then to our next presenter. And I would like to invite Leslie Acton to present to us about staking claims to ocean space through a failed large-scale marine protected area proposal that relates to Bermuda. Go ahead, Leslie.
Thank you so much, and thanks for asking me to be a part of this panel. I'm already excited to hear a lot of the things we've heard. So I always like to start off talks by letting everybody know that I'm a person who stutters, as you can see, and so if at times I'm pausing at odd places, that's what's going on. So as Emma said, my work is about a failed attempt to designate a large-scale MPA in Bermuda. And the Blue Halo was proposed in 2010 and promoted by Pew Charitable Trusts. And the idea was that it would consist of a large no-take ring around Bermuda. And the ring would start at 20 nautical miles off of the coast and extend out to 200 or the edge of the EEZ. And at that point, almost all fishing and other extractive activity that was happening in Bermuda was within that 50-mile ring from the coast. So Blue Halo proponents thought that it would be an easy win because nobody's out there anyway, then they don't really care. So my study examined the narratives that people used to promote or oppose the Blue Halo, depending on their interests. And these included narratives that the Blue Halo would stop illegal fishing or promote tourism. And those opposed talked about how it could be a hindrance to potential the fisheries in the future or to deep sea mining. Or they said that the Blue Halo is an example of foreigners like Pew trying to infringe on Bermuda's sovereignty. So all of this became extremely heated and divisive. And at the end of a very in-depth public consultation, the government announced in 2015 that insufficient information existed to make a choice. So currently there is no Blue Halo in Bermuda. So from this, it seems like then that nothing really happened. But in analyzing all of these narratives, I found these three key common underlying values or spatial imaginaries of the EEZ that both the Blue Halo opponents and proponents used in their arguments. So these commonalities do exist, and they may offer opportunities for the future in common governance options. And more broadly, these negotiations did have lasting impacts in Bermuda, even though not many people are out there, and there hasn't been any policy change that came from them. And so it's important to understand from this that even undesignated MPAs are political, and they impact ocean and social spaces. So there's my overview for now, and I'm happy to take any questions.
Okay, thank you very much, Leslie. I can see we've got some comments coming in. This is a really big topic when we come to talk about stakeholder engagement and consensus building. And now, Martin Russell from Australia is asking, do you think the objectives of the MPA proposal were clear from the start? Was the polarizing of views increased by the extremes of views?
Yeah, I think that that's a great question. I think that for this MPA in particular, the objectives of implementing the MPA to start were actually to kind of bolster support around a larger high seas MPA in the Sargasso Sea, relating to the previous panel a bit. And so I think from this international scale, where this came from, I think that the implications of what the proposal would mean in country weren't recognized. So I think that gets at your question of the objectives of the MPA were very broadly to promote conservation and then economics and sovereignty by the end. But I think because the initiative emerged through a larger conservation push, the politics of that in a small island were not taken into account from the beginning.
Okay. Arjen, would you like to ask your question directly to Leslie?
Yeah.
Make your comment.
Maybe that's easier because I was typing away, but I'm not sure whether the question is actually what I want to ask. Now, I was involved in a marine spatial planning exercise on the sidelines in Seychelles, and there was a similar polarization at the start of the project. And that caused the big tuna to walk away, to storm off, and people throwing chairs through the room, and it was horrible. But that's only a stage, in the sense that there, at least the government wanted to continue the process, so they kept pushing and pushing, and adapt or change the objectives based on the feedback, change the place where things would be protected. And in the end, I think they succeeded after I had left there. But so I'm actually wondering, because of the polarization and the struggles at the start of such a project, which can still take years to work out, whether the project aims to continue at some point and step over those discussions and work towards solutions.
Yeah, so to begin with, I can't totally answer the question as to if the project aims to continue, but I'm fairly sure from the last I knew that the Blue Halo project is done there. But I think it's really interesting. So part of the impacts of this were actually at the start, it was not very polarizing, but as it got closer to the time that a choice had to be made, and at the same time a change in the government happened, and so all these politics happened, it became divisive toward the end. And then because of that, these organizations and people on the island that had been allies for a long time at times split. So in environmental NGOs, for example, on the island, split over this, and some said yes and some said no. And at the same time, an MSP project, a marine spatial planning program, was just starting up as this ended. Interestingly, a funder of the the Sargasso Sea initiative that was kind of tied into the Blue Halo part was funding the MSP project. And because of that, it exploded because of all of this tension from the Blue Halo. And so those impacts can happen in terms of ocean the management into the future. I can't say what's happening currently, but I do know that there is a new push years afterward to implement a plan. And what that means, I don't know.
I'm just noticing that we have Joanna Pitt from the Coastal Zone Management Unit in Bermuda who has her hand raised. Joe, can you unmute yourself, or Fadila, can
you hear me?
Yes.
Awesome. Hi, Leslie. So nice to see you after a while. It's been a long time. So for a little bit of context for those who haven't read your paper, this proposal sort of, we were put in a very awkward place where we felt like we had to agree, and then all of a sudden this went from being, an MPA to, for context, this MPA would have taken up 94% of Bermuda's EEZ. And the feedback saying, this isn't going to work, 94% is too much. And then Pew went hyper aggressive on their campaigning. And as Leslie notes, it was very polarizing.
Yeah. I can see in the comments.
Her project was like therapy for a lot of us. Just being able to sort of explain how we felt, and it was everybody. So yeah, it blew up, and the government is like, yeah, we're just not going to be able to move forward in this situation. We also lost momentum on our marine spatial planning. We are currently doing marine spatial planning now. It took 5 years of recovery from this incident to actually get people to a place where we could even talk about marine spatial planning. It was that bad, and it was basically from, it was pushy outsiders. And one thing that Emma always emphasizes is like stakeholder consultation, build your local relationships. This is what everybody says. And it's like, sometimes you get lured in by all these promises, and it's just like, you have to take it slowly, and you have to listen to what's going on the ground.
So there are two other good comments in the chat messages there that, this is not anything to do with one particular place. This happens in all parts of the world. Comments about certain lobbies pushing too hard, which didn't meet the objectives of the protected areas. Another comment from the US about the importance of transferring science and other research to stakeholders. Believing in science-based management is important. Okay. This is good discussion. Thank you, everybody. We do need to move on because we do have Julian and Lisa Marie who are ready and waiting to tell us about also collaborative approaches. So can I please invite you, Julian, to tell us about your work on the gateway?
Thanks, Emma. So good afternoon to all. First, let me start by thanking you for giving me the opportunity to be here as we exhibit our resilience in these times. As said, my name is Julian Walcott, and I am the Technical Officer at the Caribbean Protected Areas Gateway, which is a regional information and resource hub, which is aimed at improving the long-term conservation and management of our natural resources, primarily through protected and conserved areas. We primarily aim to do this by providing accurate, up-to-date, pertinent data for decision making, by providing tools and services such as data mapping, reporting, and by enhancing capacities, building networks, promoting collaborations throughout the region. And we work with 15 countries throughout the region. These are the African, Caribbean, and Pacific group of states. So my summary today will highlight some statistics on marine protected areas within this group of countries, as well as some insights into the management that we kind of elucidated from some assessments that we helped to conduct. So within the ACP countries, the number of protected areas have been increasing throughout the year, but as it stands, only 5% of our marine and coastal area are protected. So it highlights the fact that there's still a lot of area that needs to be protected within the EEZs of our countries. And it's really important when we think about the shared resources, the movement of the fish stocks, the sea turtles, and the different resources and the need for protection. The majority of the marine protected areas are category 4, designated by the International Union for the Conservation of Nature. And these represent habitat or species management area. And as the name suggests, they have a primary objective of maintaining, conserving, and restoring species and habitats. Now, the majority of MPAs fall under government, so federal or national ministries or agencies. So again, it highlights the need for movement away from a top-down approach to more joint or collaborative approaches with respect to management and the involvement of NGOs, local communities, indigenous peoples. What we also found is that management plans tend to exist, but for the most part, a lot of them are outdated, and where they are dated or up to date, they're not implemented. So management plans exist, but what has been outlined in them are not being implemented. So when we think about management and the achievement of management, the Aichi Biodiversity Target 11 outlined 10% of coastal and marine areas to be conserved through effective and equitably managed marine protected areas. Unfortunately, when we look at the indicator of how this is monitored or tracked, it simply looks at the number of assessments of management effectiveness that have been completed by countries for each protected area. So it's more looking at area rather than quality. Now, of the MPAs within this area, only 36% have actually undergone management effectiveness assessment. So that highlights the fact that a lot of MPAs are not assessing management to see where improvements can be made, where changes are necessary. So this is an important fact. Another important statistic is the fact that for those MPAs that have had assessments, the majority, 60%, have only had a single assessment. One assessment basically represents a snapshot. So we therefore need to have more repeat assessments to be able to monitor and track changes and progress and be able to alter management as is an iterative process. Now, these management effectiveness assessments or PAME assessments primarily evaluate the extent to which management is protecting the values and achieving the goals and objectives outlined by the MPA. And it's based on an overall framework for assessment, which was published by IUCN's World Commission on Protected Areas, or the WCPA. And it basically takes into account 6 distinct stages or elements of management, which looks at the context, the planning, the input, the process, the outputs, and the outcome. Now, based on the assessment that we have conducted, we found that the context and the planning component of management tend to be the stronger elements for our MPAs, but we fall short in the areas of process. So identifying how we are going about management, outputs, what did we do, what products or services are we producing, and outcomes, what are we achieving. So these are some of the key areas that we are falling short. And we found that some of the key constraints, the effective management and the achievement of the outlined goals and objectives lie with the current design. So oftentimes, a lot of the MPAs don't cater for larger scale ecological processes. A lot of the boundaries are not demarcated. And so when it comes to enforcement and usage, this creates problems. Many times they have inadequate staff and inadequate budgets to be able to conduct the necessary activities. And as I said, a lot of times management plans are either outdated, or if they are up to date, they're not implemented. They're simply sitting on a shelf or in someone's computer. We also find that there's a lot of accurate and pertinent data, which is pertinent when we think about the decision-making and the policy-making environment. And we also found that there's a lack of collaboration between PA agencies. So this leads to discrepancies, duplication of effort, and overall a lack of effective management. And when we look at the databases that house the information, whether it be national, regional, or international, we need better reporting. A lot of information is not being reported. And if we really want to be able to accurately assess and indicate the status within our region, we need better reporting at all levels. So going forward, as outlined in the assessment, some of the key steps that we need to take would be, as mentioned before, the development of sustainable financing mechanisms, recognizing that financing kind of underpins all of this, recognizing that it's not a one fit. Different areas are going to require different things. And I think the current pandemic has shown really the need for diversification of our financing mechanisms. We also need to move away from single sector management to more multi-sectoral management, greater collaboration between the entities, and the incorporation of approaches such as ecosystem-based management, marine spatial planning, reef to reef, those types of approaches that look at a wider landscape perspective. Data is an area that we definitely need to improve. So we need to improve data mobilization and management throughout the region. And we need to also conduct more assessments. So for us to be able to really improve the management, we need to assess how effectively we are managing, and not just as a one-off or a snapshot, but we need to conduct repeat assessments to be able to have a better idea to monitor and track progress. We also need to periodically update our management plans. And when we do that, we then need to implement the management plans. And then finally, we need better reporting overall. So we need to better report data and information at all levels, and communication, as has been mentioned before, is extremely important. We need to be able to communicate between and among stakeholders and practitioners, and especially being able to bring and translate the science into the decision and policymaking environment, so bridging that science policy divide. So with this, the Caribbean Protected Areas Gateway will continue to strive to try to build capacity as much as possible, to try to work with countries, NGOs, agencies, to try to improve management of MPAs throughout the region. So thank you.
Okay, thanks, Julian. Let's hold comments and questions for Julian, and let's invite Lisa Marie Karuba from NOAA's National Marine Fisheries Service to tell us about protecting species and places, a partnership for place and species-based conservation. Lee.
Thanks, Emma. Can you hear me okay?
Loud and clear.
Okay, perfect. So I'm going to talk a little bit about a collaborative partnership that we have within NOAA. So I work in the Office of Protected Resources, and our office is responsible for work under the U.S. Endangered Species Act, as well as the Marine Mammal Protection Act. I'm going to focus mainly on the Endangered Species Act, though. And the reason I'm going to do that is because there is a section of that law, section 7A1, which actually is about proactive conservation. So it requires that all federal agencies in the US engage in proactive conservation of threatened and endangered species. And a while ago, we realized within NOAA that we probably needed to talk amongst ourselves. And actually, NOAA has a lot of sub-agencies underneath it. And Protected Resources realized that we could take advantage of our partners in the Office of National Marine Sanctuaries, because many of our sanctuaries actually have threatened and endangered species. They also have habitats for those threatened and endangered species, and they have management plans to try and really conserve, protect, even restore areas for a lot of really important species that our office manages. So in talking with the Office of National Marine Sanctuaries, we created this Protecting Species and Places Endangered Species Act Conservation Partnership. But we also decided that it really couldn't be just with our offices, right? It was good to try to bring in others because we share things like essential fish habitat for fishery species. Some of those designations, again, also many habitats that are in sanctuaries, also are used by listed species. So there were a lot of other connections within NOAA. And so we've brought in also programs like the Coral Reef Conservation Program, as I'm sure many of you are aware, based on some of the talks, including the talk on like stony coral tissue loss disease today in earlier session. We have coral species listed under the Endangered Species Act as well. So there's just a lot of shared resources. So we began this partnership just between Office of Protected Resources, Office of National Marine Sanctuaries. We've now expanded it to add additional offices. Now, what are the goals? So we set up early on some goals to work not only within NOAA, but also to take advantage of the fact that sanctuaries, as well as protected resources offices, have partnerships outside the US. Focusing in on the Caribbean a little bit, the Office of National Marine Sanctuaries actually has partners that they call sister sanctuaries in a lot of sites within the Caribbean. And the reason that's important is because some of those sites were set up specifically to protect animals like the humpback whale, shown here on this slide. But there are actually other resources within those sanctuaries. So if we can take advantage of the existing partnerships to actually try and expand to other types of kind of ideas about management of these animals, we can actually take advantage of the existing partnerships and potentially even expand partnerships to aim towards species recovery. So a goal of the Endangered Species Act is actually recovering species. What does that mean? It means we get to take them off the list. They're no longer threatened or endangered. It's a great goal, but it can only get there with a lot of collaborative partnerships. And so one of the things we've done internally is to look at management plans that the sanctuaries have and to look at their planning process to make sure that it's better integrated with what protected resources offices do, which are things like status reviews to determine whether or not we should list species, and then also status reviews for species that are listed to see how they're doing. So every 5 years, we do status reviews to see whether or not the condition of those species have changed. We also have started integrating that then into the planning process, but we've also started coming up with ideas for other collaboration. So outside the US, outside sanctuaries managed by the Office of National Marine Sanctuaries, ideas like, you know, other ways to think about these conservation efforts and the collaborations that we already have. So with the Nassau Grouper, that's also shown here, the things like SPA protocol, things like the Western and Central Atlantic Fishery Commission's efforts and spawning aggregation working group. So it's really this program that if you listen to the talk, has a lot more aimed at just talking to one another, right? Just sharing those ideas, knowing what each of our offices are working on, and making sure then that we can kind of strengthen all those partnerships and take advantage of areas where we can do things like cost sharing, do things like data share, do things like really better manage, because if our management plans for sanctuaries, for instance, incorporate recovery goals and actual projects towards meeting those recovery goals, we can go a lot further working together towards actually conserving these species in important places. So I'm just going to stop there.
Okay, terrific. Thank you very much, Lee. Now, let's see, there's some comments coming up about connectivity, and some comments, Ian commenting about the Dutch Caribbean Yarari Sanctuary for sharks and cetaceans, potential for collaborations there. I guess we could put this to the audience, in fact, to ask the managers who are online with us, how they're doing in terms of working with any endangered and threatened species that they're targeting with conservation action. How are they balancing ecosystem level management and monitoring with species level efforts in the Caribbean? I can see we've got a comment coming from Puerto Rico, Lisa Marie. Could NOAA recovery actions under this partnership include proposing the 2 remnant Nassau grouper spawning aggregations of the US Caribbean into spore, since we don't have a national marine sanctuary in the US Caribbean EEZ? Do you want to take that while I ask others to comment, please? Any comments?
It's a great question. So the SPA protocol can't necessarily be used to create places in US jurisdictions. So though I will admit that SPA is not my area of expertise, because I don't really work under it, it's another division in my office, but I will say that that part is true. Also, the Endangered Species Act and recovery planning cannot be used to create MPAs. It can be used to get data for doing things like designating critical habitat, which actually there is an effort underway, and it's been part of our PSP program, where we've done a lot of collaboration to help get data for designating critical habitat for Nassau grouper. But critical habitat designations under the Endangered Species Act, those can only be in US waters. So we can designate species that include foreign designations, but we don't have obviously the power to regulate what other nations do. So therefore we can't designate critical habitat. But again, with some of the sister sanctuary agreements, there is an agreement with Cuba, for instance. So that one could be used to try and help manage for Nassau grouper as well. But in terms of using SPA for Puerto Rico, because Puerto Rico is US jurisdiction, not necessarily.
All right, there's a further question there. Can I get you to take a little look for me, Lee, at Michelle's follow-up question in the question and answer box? While I look at a new.
I don't have my glasses with me, but let's see.
Okay, and just and meanwhile, a question for Julian: what methodology you use for management effectiveness assessment, and how it compares with MET, Management Effectiveness Tracking Tool assessments?
Yeah, yes, actually, the tool that we did use was the MET tool. So some of them use the advanced MET, and the last assessment used the MET 4, which is the latest version of the MET tool that's out now.
All right, so, yeah. Very good. Okay, well, we are just about coming to the end of our session. So if anybody had any further comments or thoughts to share, please flag them to me now. If I've missed anything, let me know, or are there any hands raised that I'm not seeing? Because otherwise, I would like to highlight to you that even though this session is coming to a close, there are some really interesting presentations in other sessions that also relate to stuff that's relevant to marine protected areas. On Monday in the governance session, there was an interesting presentation on alternative livelihoods or sustainable livelihoods options in the Mesoamerican Reef region. One of our MPA Connect monitoring gurus, Hazel Oxenford, had a talk on Monday also in the small scale fisheries session on FADs in Dominica. There's a couple that are still coming up. Look out for Ron Hill, who's our fisheries management guru, who's worked with MPA Connect. He's got a presentation Wednesday in the spawning aggregation session on detection of grouper spawning aggregations. There's what will be a really interesting presentation on sanitation best management practices in Roatan. That's on Thursday. Antonella Rivera. Karen Eckridge, our good colleague in Bonaire in Stanapa, she will be speaking about coral bleaching in Bonaire Marine National Park on Friday. Are there any other presentations we should mention? Otherwise, please be sure to catch the closing keynote address on Friday, 12 p.m. by one of MPA Connect's gurus and friends, Dr. David Gill. He'll be speaking about towards effective and equitable marine conservation. Okay, I'm seeing some chat messages coming in. Thank you, thank you. Good presentations. Thank you so much. Yes, it is short, but great to be with everybody. And thank you to the presenters who did all the hard work. Thank you to our panelists for taking the questions. Everybody be in touch with each other, make the most of the app to connect and to follow up and ask anything else that comes to mind. Hopefully we can all learn and take on these challenging issues such as we discussed today. All right. Thank you, everyone. Bye.
Thank you, Emma, and thank you to all our panelists today. For those of our attendees here, I just want to remind you that we have our poster session today. We have probably about close to 50 posters available to you. You can interact live with the presenters. So any poster with an odd number will be available between 6 to 7 Eastern time, and those with even numbers will be available between 7 to 8. So you can interact with posters either through the live chat, or some posters may have a live stream available for you. So thank you once again, and we will also see you tomorrow at 9 a.m. Eastern time for our keynote from Dr. Will Patterson. So thank you again, and see you all later today for the post session and tomorrow. Goodbye.